Welcome back, everybody. I hope you had an enjoyable lunch. So we are ready to get going with the second half of our mini symposium focused on veterans health. And we're going a little bit maybe beyond veterans, but highlighting a couple of the projects that are supported in our Counteract program. So this is the program that's developing countermeasures to treat exposure to chemical warfare agents. So the first presentation is by Dr. Mohammed Attar, who is a professor at the University of Alabama, Birmingham, where he serves as vice chair for basic research and the co-director of the UAB Skin Disease Research Center. Mo?
Thank you so much. Thank you for inviting me. I'm really honored to be here. Today I will be presenting some of the data with highly toxic chemicals that were developed as chemical warfare agents. So there are a large number of highly toxic chemicals that were developed during World War I and World War II, and they still are there, and they are buried or their stockpile exists, and they pose significant threat to environment and human health. Homeland Security identified 200 of those chemicals and segregated them into a toxidrome and classified them into 8 categories. So one of the category is vesicant, meaning thereby that these chemicals, when they are exposed to skin, they cause blistering, pain, tissue disruption, and highly painful wounds. So arsenicals is categorized as one of those category agents that when exposed to skin, within seconds it causes big blisters, and that's why it was weaponized during the World War I and World War II. The most effective compound as a chemical weapon was Lewisite, and its name was given as dew of death. It was in New York Times and all the newspapers that it will, if it is used, it will kill most of the populations, both civilian and military population. But thank God, it was never utilized during the war, and war record occurred. So as I told you that this causes skin, eye and lung burns, and also in these exposed populations, systemic injury also occurs that leads to significant morbidity, and if these chemicals are not decontaminated, they also lead to mortality. Molecular pathogenesis of the lesions caused by exposure to these chemicals is largely unknown, and because these chemicals are very toxic, it is very difficult to understand the molecular pathogenesis under the laboratory conditions. So we have to go to special organizations which have permit to hold, store, and synthesize these chemicals, and then we have to take our animals to expose them. That's very expensive proposition, and that's why the research on those chemicals is very limited. Now, unless we know the molecular pathogenesis, it is very difficult to develop mechanism-based effective countermeasures to mitigate the toxic effects and lethality of these chemicals. So this is the organization known as Organization for Prohibition of Chemical Weapons in Hague, and it prohibits synthesis, use, or any kind of possession of these chemicals, because despite this, these chemical weapons even recently have been used in various wars, and Saddam Hussein used similar kind of weapons. Russia used, then Assad used. This is what the news is all about the use of these chemicals. And recently, in recent conflicts, also poisonous chemicals like chloropicrin, chlorine, and white phosphorus was in the news. So if you see, these chemicals very quickly cause a lot of burns in the skin which are difficult to heal. So understanding the mechanism of toxicity of these chemicals is very important. Besides this, in the world from Second World War, the unused chemical weapons are dumped all across the world, and this is the distribution of arsenic-based weapons. And you can see that in America and in Europe and also in China and other countries, there is a strong burial or ocean dumps of these chemicals. So these, due to decay of these chemical weapons, they still pose a lot of threat to human health. These are very simple chemicals. They are derived from arsenic trichloride, and you can see that from this there are large number of chemicals can be synthesized by a chemist in the lab, and that's why they also pose terrorist risk to synthesize these chemicals and use it in civilian setting or to terrorize the area. There are various chemicals with nickname like Clark 1 and Clark 2, and they cause they are irritant, and they cause sneezing, vomiting, besides other skin vesicant actions. So what is the use to what we need to develop effective medical countermeasures for these agents? Number one is we need suitable animal models which can recapitulate the pathogenesis of these chemicals, and based on that we discover the molecular pathogenesis of lesions caused by the exposure of these chemicals, and then we look for common pathways for developing molecular toxidrome, because there are a large number of chemicals and stockpiling a large number of medical countermeasures is difficult. So we should have a common drug for the treatment of large number of similar action chemicals, and therefore we should understand the defined molecular toxidrome of these chemicals. And then it's very difficult process or long time-consuming process to develop a new drug molecule. So first objective is to repurpose the existing drugs which can mitigate the toxic effects of these chemicals, and also then develop novel small molecules with IP rights so that commercial organizations may be interested in developing those agents. So based on that, our focus was to develop murine and porcine models of exposure to these vesicants, then understand the molecular mechanism, and then do the translational aspects of drug development. So first of all, we developed murine model, and you can see that these chemicals, variety of these chemicals like leucite and Clark 1, Clark 2, they, when exposed to skin, they cause blistering in the mouse, and in histology, they produce similar lesions by creating microvesicants and inflammatory actions. Similar effect occurs in the pig model, and you can see similar blistering. Pig model is more close to human skin, and you see fluid-filled blisters in the pig skin. This is the first model that we developed showing similar kind of action as has been observed in humans. So at molecular level, the skin is similarly affected, and tight and adherence junctions are the first target, along with basement membrane, both in porcine models and in mouse models. And we have mechanistically shown that how YAP is released from the membrane as a result of disruption to adherence and tight junction, and that leads to transcriptional activation and pathogenesis of these lesions. Similarly, we also show that in mouse and pig, the inflammatory response is quite identical, and we observe both huge infiltration of macrophage and neutrophils, and that occurs across multiple chemicals and across the two animal models that we have investigated. So now the question is that there is a limited chance for manipulation at the exposure center, which we used is MRI Global, because they charge huge amount to conduct these experiments. So we wanted to develop a surrogate chemical which mimics the action of these agents, but also cause similar injury, but at a very high dose, and so that we can use in the laboratory. So we decided to use phenyl arsine oxide, which is a degradation product of these chemicals, and test that whether it produces similar effect or not. And as a result, we finally standardized protocols for phenyl oxide based injury, and we found that both histologically and physically, as well as mechanistically, phenyl arsine oxide produces similar kind of lesions that occur following exposure to other highly toxic warfare arsenicals. So using this phenyl arsine oxide, we discovered in our in vitro experiments using a panel of transcription factor based assay that a pathway known as ER stress pathway is actually involved in the pathogenesis of these chemicals, and we confirmed it both in our porcine and murine skin model that this pathway is activated, that leads to blockade of protein translation. And this all occurs as a result of generation of reactive oxygen species that leads to misfolding of the protein, and as a result this pathway, which is known as unfolded protein response signaling pathway, is activated. So we looked for agents that were available and approved for some other conditions, and two of them was phenyl butyric acid and N-acetylcysteine. Both of these were approved, FDA approved, for different ailments, one for urea cycle disorder and other for overdose toxicity of acetaminophen. So we started trying this agent because they have also properties to block reactive oxygen species, as well as serve as a chemical chaperone to block the degradation of unfolded protein. 4-phenyl butyric acid has recently been approved for ALS in 2022. So this is very interesting agent, and you see that we found significant protection against leukocyte-induced lesions by both N-acetylcysteine and 4-phenyl butyric acid. And the combination was very effective, and you see all the lesions that were produced by leukocyte exposure are completely protected by the application of 4-phenyl butyric acid and N-acetylcysteine. So this is at molecular level also reverts quite a bit. I show you limited data, but we have a huge set of data. We repeated this on the level of understanding whether it relieves ER stress, and you can see that 4-phenyl butyric acid combination almost reversed the phosphorylation of EIF2 alpha, which is an indicator of ER stress-based protein translation block, and ATF4 expression is also abrogated. The downstream signal of ATF4 is CHOP signaling, which is also protected. Then we looked in RNA-seq data, and we found that the genes which are overexpressed by the exposure to leucite are significantly reduced, particularly by the combination of leucite, by the combination of 4-phenyl butyric acid and N-acetylcysteine in leucite-exposed mice. So this was wonderful to test, to find out that this combination drug is highly effective antidote for this. Yes, sir.
How are the drugs being administered, topically or systemically?
This is a topical preparation.
Thank you.
So we did ChIP-seq assay, and in ChIP-seq assay also we found that alterations in all 5 segments of ChIP-seq, we found a quite recovery of the genes interaction, DNA interaction by the combination of leucite. And you can see the binding at ATF4, which is altered at leucite, is completely recovered by NAC and leucite for 4-PBA treatment. So, and this is the overlay of ChIP-seq and RNA-seq, and you see that the genes expression which was inhibited by leucite is recovered almost completely by a combination by NAC and also 4-PBA. So based on this, we think that this drug is very effective, so we moved to next model to test the efficacy of this drug. And in pig model, first we see that whether this pathway is activated as much as in mouse model, and you can see that there is quite overlap between the signals, particularly ATF4 and downstream signals, which we found in mouse model, and overall expression of the genes were also similar in mouse and pig model following leucite exposure. So this is the data of the combination of 4-PBA and NAC with individual arms NAC and 4-PBA in pig model, and here you can see that the lesions are almost completely gone in pig model compared to leucite alone data. And when we did mechanistically, whether mechanistically the targets are the same, and we found that yes, indeed, this combination in the pig model also attenuates the signaling as significantly as in mouse model. So we concluded that 4-phenyl butyric acid in combination with N-acetylcysteine provides a highly effective MCM for leucite and acts by attenuating ER stress and protein translation. This treatment is equally effective in other arsenicals induced vesicant injury that we have not shown here. So then at the same time when we were conducting these studies, there appeared a Nature paper in which they showed that epigenetic signaling can protect against sepsis and sepsis-induced inflammation. And the molecule that was modulator of epigenetic signaling was bromodomain 4, BRD4 molecule. So we thought that since these lesions are pro-inflammatory and they also create similar kind of morbidity and mortality as in some way occurs in sepsis. So we wanted to test this hypothesis in this model, and we wanted to see that whether BRD4 engages the pathogenesis of these lesions as an epigenetic target. So what is BRD4? BRD4 is a reader of changes in chromatin. In chromatin, when acetylation occurs, the chromatin opening occurs. This is known as chromatin remodeling. At that time, BRD4 is recruited to acetylated histones, and it leads to transcription of genes which particularly are involved in inflammation, tissue damage, and tissue remodeling. So we wanted to investigate whether BRD4 target is upregulated or not. So since then, when we were studying recently, you see this target was proved very interesting in various other diseases and metabolic disruptive disorders. So a large number of companies are developing inhibitors of bromodomain 4. Coming back to our model, what we did first, that we wanted to show that whether bromodomain targets are upregulated following arsenical exposure. And these 4 arsenical skin, we found that the BRD4 target genes are upregulated significantly in this model, and these BRD4 targets not only upregulated by these chemicals in skin but also in multiple organs, and that may be perhaps the similar mechanistic underpinning for systemic injury caused by the exposure to these chemicals. One of the key molecules that we found is disrupted is solute carrier proteins, and solute carrier proteins are involved in edema and some other solute accumulation disruptions. So this was also the major target proteins, and this is not the part of this this study further, but just to show you that we found many other targets that are involved and we are investigating. So here is the data we generated with POW and compared it with Lewisite. We see hyperacetylation of a large number of proteins following exposure to phenylarsine oxide, and we also see that BRD4 acetylates a large number of histone 3 and histone 4 lysine targets, and you see activation of these targets in phenylarsine oxide as well as Lewisite treated skin. So this proves that BRD4 is involved in the pathogenesis of arsenicals induced lesions. Then we used a classic inhibitor of JQ1 to see that if we block BRD4, whether it blocks some of the pathogenesis of arsenicals induced skin damage. And in this study you can see that when we treat JQ1 in phenylarsine exposed mice, we see protection against inflammation, and the genes which are induced significantly reversed. Out of 73, only 48 genes show reversal in the expression. Also, genes which are associated with inflammatory response and tissue disruptive response are reversed by the treatment of BRD4. We did ChIP studies to see the reversal of binding to BRD4 target is reversed by the treatment with JQ1. So we show that JQ1 is involved in the pathogenesis of arsenicals. Now the question is that whether same is the pathogenesis in porcine model. So you see These are the lesions in porcine models with leucite and different arsenicals, and this is the expression of genes of BRD4 and associated histone 3 and histone 4 transcription targets, and you see activation of these targets along with BRD4. This is true for leucite as well as for other arsenicals here. And these are the overall target genes which are involved in inflammation and tissue disruption that are induced in porcine skin by leucite exposure or other arsenical exposure. So it looks like that the pathogenesis of arsenicals is quite identical in the two models, both porcine and murine. So because we found in arsenical exposed lesions that a large area is necrosed, and the pathway that leads to necroptosis is triggered through the activation of RIP kinases. So we looked for the activation of this signaling in these lesions, and we found that indeed RIP kinase signaling is activated in leucite treated skin as well as in POW treated skin, both in murine and porcine models. So based on that, we hypothesized that if we make a molecule that engages both BRD4 and RIP kinase 3, perhaps can be a robust target for attenuating these lesions mechanistically. So we collaborated with Southern Research, which is a drug company, and tried to build a molecule. They produced 500 molecules, out of which one SR 43887 was proved to be a very effective molecule that engages with both the targets and blocks the pathogenesis of skin lesions. So this is the PK value of this. So pharmacologically this was also very good, and the toxicity was also minor. It was not very toxic in the early non-GLP toxicology studies. We show you only a small set of data here. So we treated this compound against leucite, and we found that it inhibits the target as well as it blocks the injury in mouse model. So that was very good for us. We moved to porcine model, and you can see that in porcine model also this is very effective, and it also engages the same target as we found here. So then the question was that whether same target can inhibit the systemic injury of the lesions caused by these chemicals. And this is the data from my other colleague, Dr. Ahmed and Dr. Veena Anthony, who did pulmonary studies. And here we show that in the model where we exposed with mouse with POW topically, we find impairment in the blood gas and pH in the lung tissue. So this impairment was also blocked by the compound which SR developed, which is a single inhibitor of BRD4. They are conducting studies with dual inhibitor, but with single inhibitor of BRD4, it was highly potent in blocking systemic injury. This is the chronic model. This is the model of delayed injury caused by single exposure to arsenical, and here also you see the reduction in fibrosis characterized by collagen staining and alpha SMA staining. So we think that we found a molecule and a target that is very effective both in systemic and topical injury. This is another example where we found that BRD4 inhibition can lead to blockade in kidney injury, AKI, which is induced in these mice also. So this is to show you our progress. We developed about 30 molecules which are in pipeline, and some of them in advanced stages of development. One of them is the combination of NAC 4-phenylbutyric acid, and then the other is a combination of a molecule which is a dual inhibitor of BRD4 RIP kinase, which we show efficacy in 2 animal model system, and we have all toxicity and preclinical data to support its further translation and quick development of medical countermeasure for such agents. We have new molecules also coming up in progress. These are the new targets which are recently being shown, and they are through type 1 interferon signaling, which is C-gas sting, and we found a very novel mechanism in which transposon retrovirus, endogenous retroviruses play a significant role in the pathogenesis of these lesions. So this is not the part of this talk, so we have not shown you this data, but we have something which is coming up and is very important. So in summary, we have established murine porcine models of skin exposure to single high-dose vesicants that recapitulate the human pathobiology of cutaneous and systemic injury. We developed molecular toxidrome of a blistering agent with an objective to develop single MCM for multiple toxic chemicals. We identified additional multiple druggable molecular targets and assessed their potential utility in MCM development. Then we have 2 distinct MCMs that have advanced with remarkable efficacy in 2 animal models against Lewisite and other vesicant with distinct molecular target and mechanism-based actions. We developed impressive pipeline of MCMs which are at different stages of preclinical development and include novel with IP right and repurposed drugs. So our translation approach is mechanism to clinic. In this regard, we also identifying surrogate diseases which with similar molecular pathogenesis could be used for prompt FDA approval and strong stockpiling. So thank you, and I would like to acknowledge NAEHS support. One was our earlier U54 and current U01, and then this is the big team who has contributed to this data. There are a large number of people who were associated with it. I would like to thank each of them. And then MRI Global is the place where we did most of the work with warfare chemicals, and we utilized phenyl arsine oxide in our lab. So we teamed with them to do our work here. Thank you.
Thank you, Mohammed. That was a great presentation. Questions? Daryl.
Yes, sir.
Outstanding, Mohammed. That's just, I mean, for those of us that were trained doing the classical area of protein chemistry and molecular biology, that's great. You don't see that much anymore. Did I hear you say something about nociception?
Nociception?
Yeah.
Yes. So they have great, these are the agents which cause debilitating pain in the body. And we have data in which when we apply these chemicals, we see TRPV1 related reduction in the pain. But we haven't shown that data because of the time, we focused on the preclinical development only.
Yeah. So I was hoping to see some ex vivo preparations from, some type of neuron, but I'm sure you're working on that. Good job. Great, thanks.
Yes, sir.
Excuse me, in one of your earlier slides, you showed that ROS production was one of the initial steps in the pathogenic cascade. Have you defined what the enzymatic and cellular source of that ROS production is?
So we found two molecules. One was initially xanthine oxidase that was activated, xanthine oxidase. And then there is another molecule that in lungs, Veena has identified. We published in paper. It is a, I'm skipping the name, but that is also involved in the oxidative signaling. Also, in some of the studies, we found that the production of oxidative stress is through the quinone reductase. Quinone reductase reduces the quinone levels into hydroquinone, and in an environment where there is a damage, this hydroquinone is recycled into quinone, producing reactive oxygen species. So, there is a complex induction of various mechanisms by these chemicals through various pathways. Yes, sir.
I concur that this is a beautiful, large body of work that you discussed. So as far as the follow-up question here, the oxidant stress, couldn't some of this be coming also from mitochondrial damage? That would also help link up an explanation for activation of the gas-sting pathway and other things that you were talking there about, and that would imply some broader systemic effects through that type of mechanism.
So these chemicals are devastating chemicals, and if you see the damage both in humans and in animals, it's huge. It leads to release of both mitochondrial DNA as well as nuclear DNA. And that part that I'm showing, I did not show you, and I told that I'm working on C-gas sting pathway signaling and type 1 interferon induction is related to the sensing of those DNA fragment, in which you see the release of endogenous retroviral fragments, and as a consequence, those fragments expectate with other genes, and they carry the regulatory signal. So the expression of genes which are involved in inflammation and tissue disruption is several-fold higher regulated through the incorporation of those signals.
Well, one more question.
Sure.
Regarding your treatments. When did you have to give those treatments, and are they effective once the damage occurs and even the long-term consequences you're showing?
So these are the topical agents that we are using. We can use them multiple times topically, and in our protocol we have tested them between 30 minutes to 60 minutes of exposure. Because each experiment, if we do more like 2 hours or 3 hours, for each similar replicates have to be run with lewisite and other arsenicals, and they asked us as much cost as $1 million just for exposure. It was practically impossible to run with MRI Global, I must admit. So these studies are cost prohibitive. Yes.
Very nice presentation. As you know, BRD4 is a member of a large family of BET inhibitors, and so I wondered if you looked at other members of the family and you know anything also about interactions with other epigenetic modulators.
Right. So when we started, most of the knowledge was limited to BRD4. And BRD1, BRD2 were also known. BRD7 and BRD9 were also known some way. But BRD1 and BRD2 were having the overlapping effect, but there was no specific inhibitor with which we can delineate that whether they are redundant or they are really playing a unique role. So we didn't do at that time. Now the tools are available, we are thinking. Now during this period, recently there was a science paper from AbbVie, I think. They have identified BD1 and BD2 domain. One of them is involved in proliferation and the other one is involved in inflammation. So they are looking with different models of inflammation, and they found that BRD4 is effective. Recently, some data has been shown that BRD4 is also effective against COVID-19. So, I mean, there's a lot of interest in this molecule, and I think that topically, this is very safe also compared to systemic toxicity, because BRDs have physiological role. Also, in cancer, we found BRD7 and BRD9 playing a significant role. This study was done with my mentor, Dr. Bickers at Columbia University. So we have data, but we didn't do those studies with these chemicals. Yes.
Yeah, I have a question. So I don't know if you're aware, we celebrated last year the destruction of the last of the stockpiled chemical warfare agents in the United States. And so the Centers for Disease Control and Prevention actually was in charge of the protection of the public health and the workers' health at the facility, in charge of the destruction of the over 30,000. But so this is the good news. But what has happened in the original map when you were showing, there were like chemical warfare agents that were sunk in the ocean, and now fishermen are moving, farther and farther and encountering them. Yeah. So I was going to say this is like fantastic what you're doing, and if you actually have done some research on those.
Europeans have a lot of projects because it has been 75 or 80 years and they are decaying. So the fisher, when they are being exposed. And the purpose to show that graph was to overlay with the arsenic exposure of those areas, because the degradation of these chemicals lead to enhanced concentration of arsenic, which is a measure that how much they are being released. So the question was that whether those waters contain arsenic level or not. So we showed that where these chemicals are dumped, their arsenic level is low, and the arsenic level high is in different parts of the world. There is no evidence of burial or dumping of those chemicals.
All right, thank you very much, Mohammed.
Thank you, David. Thank you.
So our final presentation in this mini symposium is also from the Counteract program and brought to you by the Miracles of Modern Technology. Even though she's attending a conference in Europe, she was able to join us for the day to give her presentation from Europe. So appreciate you staying up late to give your talk. So this is Dr. Olivia Veras, who is a professor at the University of Colorado, where she serves as the director for the Center for Advanced Drug Development, and she'll be talking about work that they are doing on pulmonary exposure to threat agents. So, Olivia?
Thank you very much for that very nice introduction, and also thank you for inviting me to give a synopsis of our research over the last almost 20 years at the University of Colorado. I'm going to be talking about the adverse pulmonary effects of highly toxic chemicals, and I will compare and contrast 3 of them in particular that we have been working on. So first I will review sulfur mustard inhalation injury, which causes mostly airway burn and alveolar fibrosis later on. I will also review methyl isocyanate inhalation injury, also causes airway burn and alveolar fibrosis. And then I will talk about chlorine gas inhalation injury, which has more mild alveolar flooding early on and also airway burn. So in terms of historical context for sulfur mustard, to start off with that, sulfur mustard was first developed in 1822 by reacting sulfur dioxide with ethylene. Shortly after that, in the 1860s, it was also developed in Britain and Germany. Sulfur mustard was the first vesicant agent that was ever used as a chemical weapon, so it makes it very long-standing, and there is lots of literature on this agent. Other chemical reactions were also developed to produce a more potent sulfur mustard over time as years progressed, more in the middle of the 20th century. Some of the examples of sulfur mustard uses as a chemical weapon were in World War I in the UK against the Red Army in 1919. It was used by Spain and France as well, Italy, Soviet Union, again, Italy and Japan, and most recently in the 1980s, the Iran-Iraq war. There have been a lot of deaths and a lot of exposed veterans as well as civilians who have been followed a lot over the last 30 years. So we have been learning a lot about them. And then in 1995, Sudan again used it in their civil war. Modern use of sulfur mustard has been, and its potential threats that we could be exposed to is, as we were just discussing with Dr. Attar, that a lot of these ammunitions were dumped into the Baltic Sea after World War II. In 1966 to 2002, lots of sulfur mustard weapons have been found by Bornholm Island, and there are many more locations that are unknown. So a lot of fishermen do seem to accidentally pick these up and get accidental exposures. Unfortunately, these weapons still seem to be very potent and very lethal, so that makes it a health concern for the general public as well. Most recently in 2010, there was a New York fisherman. There were a lot of publications of that person, and I believe there were 2. And then there have been storages as well. You can see this picture here. This is sulfur mustard storage that has been in Pueblo, Colorado, that has been decommissioned as of earlier this year. So we should not have any more, but there was a lot of leakages previously. So by 1970s, and Dr. Attar also touched on this, there has been prohibition of utilization of toxin weapons and stockpiling of bacteriological, so mostly biological toxins. But that particular convention and prohibition document did not prohibit chemical weapons. So in 1997, the Chemical Weapons Convention was put into place, the CWC, which prohibits stockpiling and production and use of chemicals for chemical warfare agents, specific ones. Unfortunately, it has now just recently been proven that in 2015, just very recently, sulfur mustard was used in Syria. OPEC and many other agencies have been analyzing a lot of samples from the earth, from animals, as well as from humans, and this has now been confirmed. So we still get exposed, unfortunately, and in particularly our soldiers in the military and when they go into these areas. So I wanted to, because I'm a clinician scientist, I wanted to start with a clinical case presentation so that we can kind of get a feel and a sense of what sulfur mustard does and how it progresses. So this particular case is from a 21-year-old from World War I, and this case was published in 1920. The reference is on the bottom. There was a premature burst of mustard gas munition during firing, but there was no immediate symptoms that he had. And you can see this is what the bunkers looked like that they were exposed to as they were trying to fire these sulfur mustard bombs. This person slept in the proximity of the artillery gun, and he awakened after about 4 hours with lots of symptoms. He had nausea, burning of the eyes, he was vomiting, was dizzy, had a headache, and he had a little bit of a tickle in his throat. Now, this continued, and within 24 hours, all these symptoms worsened. He had conjunctivitis, so redness of the eye, and that's what this looks like, with photophobia, and that's when that is having problems with light, sensitivity to light. His throat was very red and very sore. He had a hard time breathing, so he had dyspnea. His chest was rattling and he was wheezing. And he had slimy, foamy sputum that he was coughing up. He became febrile and became very weak. Within the next day, his temperature continued to be high. He became cyanotic, his pulse went up, became very restless. On the third day, this continued, and then he was not really able to be awakened much. And on the fourth day, he died after having shortness of breath and high heart rate. Now, I have to say this is in World War I, so that we didn't have any mechanical ventilation. There was no ICU. So this patient was treated, and there was no antibiotics either. So this patient was treated just with what they had, which was just supportive care, which was very minimal back then. His diagnosis was Croup inflammation, so just like croup of the pharynx, larynx, trachea, and large bronchi. He had purulent bronchitis, bronchiolitis, so these are airway problems. He had hemorrhagic bronchopneumonia, and that's infection of the alveoli and the airway with some bleeding into it. He had some bullous emphysema that occurs after blockage of an airway, when airway gets trapped in the lung. And he had some minor hemorrhages in basically all of his organs. So here's a nice picture on the right side of the entire respiratory tract. And as a pulmonologist, I think of, we have to think of every single part of this system and where injury can occur. And it really all depends on deposition of particles and severity of the inflammation and host response. So his autopsy showed that his nasal cavity, so going from top to the bottom, his nasal cavity was filled with purulent mucus. His throat and the back of his throat, as well as the larynx, so the voice box, were all filled with purulent material, very yellow, adhering and coating everything. His epiglottis and soft palate all had this membrane. This pseudomembrane is what they were called at the time, coating the entire upper airway. Now going down to the trachea below the vocal cords, this continued. There was firm, loosely adherent pseudomembranes. Mucosa was very red. And histologically from this particular patient, this right here is what you see is this fibrinous pseudomembranes that was blocking the airway. B right here, you see that is the epithelial layer, still intact, and C is the cartilage. Going down lower in the respiratory tract, so now we're at the main stem bronchi and the lobar bronchial branches. We had a lot of thick mucus plugs, and the bronchioles also had the same. anterior lungs had these blisters or bubbles that were hyperinflated, and the pleural surface of the lung also had some small hemorrhages. Microscopic finding confirmed the gross findings and shows this fibrinous leukocytic exudate and edema that happened around the airways. So that was from World War I. This particular paper is called the Willems Report, reported on patients from the 1980s Iran-Iraq war, when the Iranians were evacuated to several European hospitals for care. Those were the severely exposed patients. And they were sent to Germany, to Sweden and Denmark and Belgium as well. And so from all of these patients, this is a cohort of 65 patients that were severely impacted, and they arrived to these hospitals about on day 2 to 5 of their exposure. Unfortunately, they had 15 out of the 65 that had these casts, and 50% of those died who did have it. If these pseudomembranes, and I call them casts now, that's the language we use in the clinical world for them. These pseudomembranes also were present in some other patients who didn't die but needed an emergent tracheostomy to open up their airways. So the morbidity and mortality of these fibrinous blocking pseudomembranes is very significant, even now, as well as in World War I. So the acute phase effects of sulfa mustard inhalation does depend on a sulfa mustard dose. The individual's characteristics, temperature, and humidity also changes how the body responds to it. The main feature of sulfa mustard exposure is there's a completely asymptomatic incubation period. It could last hours, it could be months, after a very small sulfa mustard exposure, and the patient doesn't know they actually were exposed. And then months to years later, they come down with lung fibrosis and other complications. So the acute effects, again, acute airways edema, inflammation, destruction of airway cells, and these pseudomembranes are a big problem, and they cause the acute deaths. And if you see a normal airway, it's nicely open, nice lumen inside. If they're inflamed airway with these mucus or pseudomembranes in it, they just get completely blocked. And these casts or pseudomembranes, they're very firm. You can pull them out and they look like this. They look like a tree branch that basically that's why we call them casts, because they look like somebody casted the entire lung. Now, sulfa mustard can also cause multiple organ dysfunction syndrome, so bone marrow suppression, GI problems, as well as brain injury symptoms acutely, as well as later on with some neurocognitive decline and early dementia as well. So we wanted to model this exact scenario, and we started with a rat model. A rat model is an intubated, sedated model. They get exposed to sulfa mustard vapor to try to mimic the human scenario. And we then evaluated them at first at 24 hours. So what we found is that we did have a dose-dependent survival. This is a survival graph over 24 hours and percent survival on the y-axis. Based on just minute changes of dose of sulfa mustard from milligram per kilogram, we have a very significant change in acute mortality within 24 hours. And all of these animals had severe obstruction of their airway with these casts. Oxygen saturation, if we look at that from a more spread out sulfa mustard dose titration study, we could see that we have a dose-dependent saturation by 12 hours. So we do have cyanosis at the higher doses of sulfa mustard, which is shown here in purple and green. And we don't have severe cyanosis yet at 12 hours at the lower doses. So it's definitely a dose-dependent phenomenon. And what we found is that these animals, when they die, they have these airway casts in them. And this is an airway opened up during gross necropsy. And you can see this white cast here sitting inside of the lung. If we take that out, we process it for histology and look for specific immunohistochemical antibodies for fibrin, as shown here on the right upper hand corner. You can see that inside this lumen, which is lined by the airway epithelia here, is filled with this very brown fibrin positive material. So this is this fibrinous cast, and this is IgG control. So next, what we did is we looked at regionally where these occur. Where along the entire lower airway tree do we see these casts? So here on the left side, you see the cast airway obstruction % per airway region. So we looked at all of these airways from left all the way going to right on the x-axis, is basically going from top of the vocal cords going down distally into the airway lobes, lobules. And so what you see here is the higher the obstruction, the higher the cast material. What we saw is we saw a lot at 12 hours at the highest dose of sulfur mustard. We saw lots of cast within the right lung, and also in the left lower lobe, which is this very large lobe that the rat has. But we didn't see that much up at the higher subglottic area or trachea one. So it definitely goes pretty deep, but it stays in the airways. Now, if we look at this across 4 different doses of sulfur mustard, so I would call this the lowest on the left side, all the way up to the highest. And this is evaluated, I believe, at, I have a little bar here, so I have a hard time seeing it. Okay, yeah, 12 hours. So this was at, looking at 12 hours. What you see is that the 2.0 milligram per kilo sulfur mustard really doesn't have much cast % obstruction at all. That's evaluated in cross-section at each of these zones through acute dissection during necropsy. So, but we here in the right lower lobe, we have 26% obstruction. So that was the worst. If we go up a little bit on the dose, you see it's 51% obstruction, 89 at 3.0, and near complete obstruction at 3.5. So we do have a dose-dependent cast formation happening, which explains why the acute deaths that do happen only happen in those who are who received the higher dose, or they were closer to the source of the sulfur mustard. So next, we developed a quantitative system to evaluate all of the airways put together. And we use the NIH ImageJ program to help us with this scoring method development. So we gave a 0 to 7 score based on % obstruction at each of these 5 regions, as shown on the left side picture with these black lines. And then we weighted them based on % volumetric use or volume from each breath into each lobes, and got a composite score. So on the right side here, you see a composite score for all those 4 doses of sulfur mustard, 3.5, 3.0, 2.5, and 2.0. We evaluated animals at 4, we euthanized them at 4 hours to evaluate for cast, and we saw none in any of the animals. But by 12 hours, we have a very high high amount of cast in all, putting all of them together into one score at the highest dose of sulfur mustard, which dose dependently decreases. So we also evaluated these animals by CT and tried to see where these fibrinous clot looking casts are coming from. So here what you see is a barium infusion study. On the left-hand side is a naive normal rat. On the right-hand side with the B is a sulfur mustard exposed rat at a very high dose, and this is at 5 hours after exposure. And what you can see is we put barium into the pulmonary artery to see if there's a problem, a blockage, or why would we have clot formation in the airway? Are the vessels involved? So what you can see here on the lower portion is we have lack of arboration. So we lose some of the branching of the animal's pulmonary circuit. And that would definitely result in hypoxemia. And then we quantitated that here on the right side, and the sulfur mustard CT quantitation of volumetric quantitation of these pictures show that there is a significant decrease in pulmonary artery and arterial blood flow and volume. Histologically, we picked up significant clot formation, as shown here with the red arrow, versus no clot in the black arrow, as it does not stain for fibrin here, very fibrin positive in the red arrow. So we do have some involvement, and we do have some thrombi formation, microthrombi, in these animals that may contribute to these pseudomembrane formation. So next, we have so far, all of the work that I just showed you was funded through the NIH Counteract program, starting from 2005 through a U54 mechanism, and 2 or 3 cycles actually. And then we went to BARDA, Biomedical Advanced Research and Development program through the HHS to fund our pig model development, and to see if this holds true for a large animal as well as for the rat. So what we also wanted to see is what happens if we look at these pseudomembranes or casts with a bronchoscope. We could live take a look, see what they look like, and if we use any therapeutics, we would be able to tell on that patient or that pig if our therapy was successful. So we use this bronchoscopy tower, which is the same one that well, not the same one, but a similar one that we use in the clinical world for our pediatric patients in the pulmonary clinics. In terms of a quick anatomy, you can see this is what a bronchoscope would look at. All of this pink things would be your tracheal rings, and we're looking down the trachea now. You can see here a left main stem, so the left bronchus branches off, the right main stem branches off, and in pigs we have this little what we call tracheal bronchus, a right upper lobe that comes off the trachea. Humans don't have this, but this is what a normal pig would look like. After sulfur mustard, at 10 hours, when we developed our model down at Lovelace Research Institute, this is what it looks like, and this is what the pseudomembranes that they were talking about looks like, and you can see how thick that actually is, and I will show you a nice video in just a second. Now, if we take those lungs, fix them, we can also see and pull out these cast materials from these airways that are completely blocking it. This one here in the middle, you can see a complete occlusion by these casts. And again, they look like little branches from a tree when you were able to pull out the whole thing. We looked again, and yes, they are fibrin positive, as you can see on the bottom right hand corner here. In the bottom left-hand corner, this is a mucus stain, and what is with a H&E back stain, what's very important here is that the mucus content, which is purple, is very minimal. So this particular cast in this animal is extremely fibrin positive, but it has very minimal mucus. So any treatment against mucus will not work in this particular fibrin casts. So this is just a quick slide on how does a thrombus form. These casts are basically a fibrin thrombus. There are two intrinsic, extrinsic pathways, if you can remember back into your science classes, very complicated. So many proteins come together and get activated until fibrinogen gets cleaved to fibrin, and then it gets cross-linked and makes a fibrin clot. There are three types of clots that can form. One is for hemostasis, which has mostly red blood cells in it. That's if you have a little cut in the vessel. You can get an arterial thrombus, which is mostly filled with platelets, as well as these foam macrophage cells. And then you can have a venous thrombosis, which is a mix of both. And so what we were proposing to what usually happens in a vascular clot is you have an endothelial cell injury, von Willebrand factor gets expressed, platelets are activated, they get recruited, get aggregated, thrombin gets activated, and then fibrin comes in, and as shown here in the black little lines in this venous thrombosis, and forms a thick permanent clot. So what we wanted to do is get rid of these clots and casts. So what we use for that is alteplase tissue plasminogen activator, or TPA. It is used for myocardial infarction, strokes, pulmonary embolism as an acute fibrinolytic drug. And we wanted to test it by delivering it directly into the airway to see if we can save our animals and get rid of these casts. We developed, again, we first started with a rat model, and we decided to use a trigger to treat criteria and wait until the animals started becoming hypoxic and desaturating less than 85. And then we gave them TPA at the doses that's shown here, and gave it every 4 hours due to its shorter half-life. So what we found is following these animals out, these are rats, to 48 hours, is those animals that got TPA after sulfa mustard, they all survived to 48 hours. Meanwhile, all the animals that got sulfa mustard and all the animals that got PBS, which is our vehicle or placebo, but died by 48 hours. We do have a couple of stragglers here in the PBS group, and that is because it is possible to potentially wash out these casts and get them dislodged a little bit so the animals could mobilize them and allow some survival longer. Clinical distress score. This is a scoring method that we developed to look at work of breathing distress as well as activity. And with TPA, we have the lower the better. Clinical score, TPA definitely helped decrease the stress of the animals. In terms of oxygen saturation, it was incredible. As you could see with the red line, our oxygen saturation continued to be near or above 90%. And this is going out to 48 hours. We have now gone out to 72 hours as well. And this continues to be the same versus those animals that didn't get a dose. They all died and were very hypoxic as they did so. The CAS scores, again, were very low after TPA when we did the composite CAS score from 0 to 7, 7 being 100% obstructed, versus both of our controls. With sulfa mustard, one is non-treated, the black, and the PBS placebo one had very high CAS scores. So then we tested this in the pig as well. We did the same thing. We exposed the animals and gave them alteplase when their oxygen saturation dropped below 85%. And we were only able to follow these animals because of IACUC procedures to 12 hours awake versus the placebo, which is shown here in blue. Sulfa mustard only is in black line for survival. All of our controls died, and all of our, well, 3 N of 3, these are pigs, so we cannot use too many of them. We had good survival in 12 hours. Oxygen saturation, you can see with the saline placebo, we have a very big dip, decrease. We were trying to resuscitate this animal here and giving breaths of oxygen, but that didn't help, versus the TPA animal did pretty well by 12 hours in terms of oxygen saturation. Looking at the gross pathology here, on the left side, this is a placebo animal, right upper lobe. You can see there's a white cast material in there that is absent in the TPA treated pigs. And if we look at the cast percent of airway lumen obstruction at all the different regions, blue is in the saline, so the placebo animals, we have very significant obstruction in those at time of death versus the 12-hour animals, the TPA, they had 0 casts or in the left upper lobe. I needed to improve my technique to get rid of the cast over there as well. We delivered TPA via bronchoscope in these pigs and squirted them directly, the TPAs directly on top of these pseudomembranes. So here's my promised video, and this is the normal. On the left side, again, you can see the left main stem, right main stem, and the right upper lobe. And this is our sulfa mustard exposed animal at 10.5 hours. You can see that you can't see most of the structures. We don't even know where you really are. All of this thick white things are obstructing the airways. There are bubbles happening because they're trapped. Air cannot move. And everything looks really raw and red and very small. And so that's, and you can see it's kind of pancaked, flat, swollen. And this right here is your right main stem, which is what's shown here. And this is the left main stem, which is shown here. And this is the same animal. That's the naive for that animal, or the pre-bronchoscopy. Now, if we give them TPA, this is 30 minutes after the TPA was delivered to this pig. This is what it looks like. That white stuff is now gone. You can see it's liquefying, it's mobilizing, air is going through it. We still have some redness of the airways and some swelling is obvious, but we are now mobilizing and able to breathe. And that's really the goal, is to save the life of the soldiers or civilians or anyone exposed to these mustard compounds. So that's the sulfa mustard, but sulfa mustard is not the only agent that causes these pseudomembranes. Methyl isocyanate also does that. So we went to study methyl isocyanate and see if this treatment would also work in terms of methyl isocyanate exposure. And I will call it MIC from here on out. MIC is important because it was the cause of the Bhopal disaster that happened in 1984, where 30 to 40 tons of MIC leaked from this urban carbide pesticide plant in India, and 4,000 people died overnight, and another 10,000 died over the next days to weeks. Necropsy findings on these patients, this is really hard to find, but the few that I did find show obstructed airways, inflammation, hemorrhage, acute bronchiolitis, and fibrinous exudate in the entire respiratory tract. And that sure sounds just like the pseudomembranes and casts we saw in sulfa mustard. Methyl isocyanate is usually used in industrial manufacturing. It's very easy to make as well. So it is a potential chemical weapon, but it's also an industrial accident agent. Carbamate pesticides and herbicides are made from it. Plastics, paints, adhesives, rubber, also other isocyanates in industry. So it is used quite a bit for these processes. So here's another patient case. This particular patient was exposed to methyl isocyanate. She, female agent, age of 26. There was a cloud of gas from a nearby chemical plant. She had immediate eye burning, watering, and a runny nose. By 4 hours, she was coughing, and it was found that coughing after MIC exposure is very strongly associated with mortality. If they don't cough, they usually will survive. Within 8 to 12 hours, she had diarrhea, shortness of breath, vomiting, wheezing, respiratory distress. By the second day, she had more cyanosis, more coughing, and she was coughing up these thick, frothy sputums and had severe respiratory distress. And by day 3, she had cyanosis, seizures, frothing at the mouth, and also has died, just like our sulfur mustard patient we talked about earlier. The autopsy on this patient shows severe epitheliofibrinous necrosis, sloughing, pseudomembranes, pulmonary edema, airways edema, inflammation, and signs of acute respiratory distress syndrome, or ARDS. Cause of death was deemed to be asphyxia due to respiratory failure. So we went and tried to model this with the help of MRI Global, contract research organization, and they have helped develop this. We got some histology after exposure, and here is a trachea with a fibrinous material inside it, very similar to what we've seen in the sulfur mustard. And if you go lower into the accessory lobe, I can tell you that this is an airway that's filled completely with this fibrin IHC positive material that is the cast that we have seen also with sulfur mustard. We have done, and this was all dose responsive, so dose dependent, 0 milligrams per meter cubed versus 2,000. We did have a dose dependent survival. And if we broke it down to further to try to develop an LD50 model, which we did for the 250 ppm, this continued to be very dose dependent. So we did the exact same treatment with alteplase into the airway in the rats as well. And this was Dr. Carl White's work through also an NIH, the U54 program. And we indeed saw, this is the gray solid line, that TPA did help, or alteplase did very much help survival in these animals compared to controls. But it wasn't perfect. And this is out to 24 hours. Now it's, MIC is not sulfur mustard, but it does similar damage. But it also has a lot of hemoglobin binding, and it impacts hemoglobin oxygen carrying capacity. So it's not just how sulfur mustard works, which of course is also complicated with a lot of DNA alkylation. Either way, alteplase worked some. And when we looked at histologically, this is methicillin, or MIC on the left side, subglottic trachea and lower bronchi versus MIC plus alteplase. You could see that we were able to clean out the airways very nicely. Now, if we thought about maybe the use of Mesna. Mesna is sodium 2-mercaptoethane sulfonate. It is a thiol, and it is used as a chemoprotective agent to detoxify metabolites from chemotherapy agents such as cyclophosphamide. It's also an antioxidant, and it can scavenge some reactive oxygen species. So what you see here is when we gave Mesna with methyl isocyanate every 4 hours, those are these dotted pink lines, we changed maybe or delayed the death of the animals, and it was significant, but it's still not really great, and we were not able to survive all these animals. So we thought about maybe combining them. Can we combine TPA into the airway and Mesna, which was given intraperitoneally, so systemic drug, and see if that improves the situation better? And it did. And right here is the green line where you have both of them as a combination. And we did have much better survival to 24 hours. I mean, there was 0 survival with the MIC alone, and we had about 80% survival with the combo treatment. So that was really nice, and we think this could work really well. But in sulfur mustard to see if that would help. Well, lo and behold, when we did use Mesna and just gave it 2 hours post-exposure and 1 more 4 hours later, so only 2 doses in the beginning, we were able to delay the death of the animals. This is a rat model in an LD 100 model. So this was Mesna was improved. It has improved the survival. It also improved oxygenation with sulfur mustard. So it seems like it decreased the dose effect, the dose injury, and it also improved cast obstruction. But it wasn't. It was only temporary to 9 hours. So it sounds like we need to optimize this more and do some redosing to further improve that. Now, chronic effects of sulfur mustard, there are many of them as well. Prevalence is very important, and I think veterans in particularly would be very important. This is very important for veterans' health. In terms of pulmonary, 50% of patients do get bronchiolitis obliterans. This is airway fibrosis. They can get pulmonary fibrosis as well, and the CT scan looks hazy as shown. Their airway function, pulmonary function is very obstructive as well as restrictive, so showing that it's a mixed disease. And they can get chronic bronchitis, lots of pneumonia, COPD, floppiness of the airway, emphysema. They can also get these stenotic airways where they are just fibrosed down, large airways, very difficulty breathing like that. And they can have the opposite, which is bronchiectasis, which you can see here in the CT scan, 3D rendering. It's actually an extra opening and floppiness of and dilation of that airway. So we also developed that model in the rats and saw that we have this 2-wave of hypoxemia. We have this wave in an LD50 model, LD50 at 28 days. So those animals that survive the first week of injury, they start having progressive hypoxemia, very specific and consistent with chronic pulmonary injury. Acute injury, there is an improvement phase, and then chronic deterioration. At 28 days, when we look at them, we have significant fibrosis, as shown here, greater in the dependent lobes, and it's very patchy and heterogeneous, very similar to IPF. Airways can be fibrosed as such, doesn't even look like an airway. It's hard to figure out what that is, but it's a lot of collagen has been deposited. When we look at airways resistance, so pulmonary function, resistance goes up by 21 days and stays up. So we have also have that model developed to study chronic lung diseases. We tried nintedanib, which is a FDA approved for IPF treatment. It is a tyrosine kinase inhibitor of PDGF, VEGF, and FGF. It inhibits fibroblast migration, proliferation, and again, it is FDA approved. So what we found is when we gave nintedanib to these animals every day, oral gavage, we were able to very nicely fibrosis formation, their total lung. So this is a potentially great therapy to use in our chronic lung disease patients after sulfur mustard exposure. Now we also have late effects in the heart. We have pulmonary hypertension, right? Indeed, we have higher mean pulmonary pressures in the sulfur mustard animals. we have higher pulmonary vascular resistance, and we have found in the hearts that we do have a little bit of fibrosis. So maybe they have had some cell death and around 2 weeks after the injury. Now, chlorine gas, I won't go too much into because it is very different. But it has been used very significantly in the most recent wars in the Middle East. It is a oxidant, highly reactive oxidant, and it reacts with water. It's used in industrial doses, definitely as a chemical weapon, and it has been used, as you can see in the global attacks map on the left lower corner, in so many different regions around the world. And it causes very significant respiratory distress in the first few hours to days. The time course for this one is very immediate. The symptoms usually resolve within 6 hours, but it could also deteriorate. Deaths are usually low, but there's also a dose dependence. In terms of late effects, there is usually not very much late effects from chlorine. There is some reactive airways disease for a while. A few sporadic bronchiolitis sibilans has been shown, but again, there's a very long-term neuropsychiatric and neurocognitive dysfunction that's being described now in many, many patients. In our chlorine model, it looks like this. Airway histology is at 1 hour, very swollen. It gets more swollen by 6 hours. By 24 hours, all that swelling goes away. It gets sloughed off. When we look at their brains for neurocognitive dysfunction, we see a lot of brain dysfunction as shown in the black lines. Within 30 minutes, they have significant encephalopathy, which correlates to poor cognition. And it stays all the way through 28 days, although it does improve compared to naive, which is shown in green. Latency to fall, and that is on a rotarod, is also impacted in these animals, and they just don't seem to remember, so they have some memory problems as well as some motor function problems throughout the entire time after exposure. Now, if we give them scopolamine or atropine, we are able to improve this encephalopathy and prevent this this lowering of the EEG. And if we give it in combination, as shown in pink right here, we were able to normalize all of the power, the EEG power in these animals after dosing. And first dose happens here, and the second over there. And it is protective for the rest of the time of the study. Latency to fall was not perfectly improved, and adhesive removal time. So current projects, I'm just going to go through this. It's not that important. In summary, for acute sulfur mustard, we see lots of airway cast, and we have developed alteplase and working on Mesna now. For the delayed sulfur mustard with severe fibrosis, pulmonary hypertension, and RV dysfunction, it's nintedanib, and we're working on some cardiac medications now to test them. For the MIC, it's alteplase, Mesna. MIC delayed, we have not studied. Acute chlorine, we have scopolamine and atropine now for neurotoxicity, and it also improves survival. And for delayed chlorine encephalopathy, scopolamine and atropine work really well. And that is all of my presentation. And thank you for bearing with me with my poor connection here. And this is my team in our Center for Advanced Drug Development. Thank you.
Thank you, Olivia. Very, very compelling presentation. So we'll take a few questions for Olivia. Tim.
For I think it was the mustard sulfur gas toxicity, when you give the clot buster, what is it?
TPA, alteplase?
TPA, yes. How many times do you have to give, would you anticipate you have to give that in correlation with the ongoing toxicity of the toxin? And do you
Yeah,
get,
and we are
and do you get
developing, yeah.
any hemorrhagic complications?
So good questions. We have been doing this study with Genentech, whose drug this is, through the BARDA program. And what we are finding is by 7 days, the casts stop forming. So we expect that we would have to give it for 7 days, the TPA. Initially, the formation is faster than later on. So when we have done that study as well separately of as needed TPA, it looks like we just need to give it every 4 hours for the first 24 hours and then once or twice a day for 7 days. And do we see hemorrhage? We have not seen hemorrhage, but we have tested heparin into the airway as well. And we saw hemorrhage in 25% of the animals. So we decided not to pursue heparin. What's really good about TPA, it's very the half-life is very fast. Intravascularly, it's 1.5 minutes. Through the airway, when we give it, it looks like it is about 2 hours. And then it's out of the system.
Thank you.
Any other questions for Livia? I was also going to ask the repeated treatment question, so thank you, Tim, for getting that.
And I should mention that based on these findings that we developed through the animal models, we are able to use TPA and develop a guideline for treatment in our clinical world. We do see cast formation in congenital heart disease patients from time to time, and we have not had anything to treat them with either, but now we do. So we've developed a nice guideline, and it works to have a nice wean off the every 4-hour dosing.
Thank you very much, Livia. So let us thank again the speakers, not only Livia, but the other speakers in this symposium.
Thank you.
And so normally we were going to have a council discussion to kind of have some bigger picture discussion of where we see opportunities to leverage the veterans activities, the counteract kind of activities to advance the mission. But I think, given that we're a fair amount behind schedule, and Rick and I both have a lot of content in our presentations, I think we're going to skip the council discussion here and move ahead with Rick's presentation. And then at the end, when we have the council discussion, there's one specific topic that we want to bring to your attention, but we can also have some of that conversation in the second council discussion. So Rick, podium is yours.
So let me update you on some of the things that have been happening here at NAEHS since the last time we were together in, I guess it was back in February. Let me have the first slide, please. Here we are. Well, let's talk about budgets. So that's happened since the last time we were together. So Congress actually did pass a budget so that we actually know how much money we have to spend for fiscal year 24. So the good news is that, well, for FY24, flat is good. So unfortunately, we didn't get the types of increases that we had come accustomed to over the past several years. So looking at the second bullet point here for the labor HHS, which gives us the bulk of our funding at NAEHS, we were flat from FY23. So of course the cost of doing science goes up, the cost of personnel goes up, so we're not able to do as much as we could in FY23, but nevertheless, again, it's flat was good. There were some programs across the NIH that didn't do so well, that they were actually cut pretty significantly. There's some other programs, for example, at the, actually Gary and I were at a meeting last week, so the National Institute on Aging actually had a considerable increase, well, I guess not a considerable increase, an increase to study Alzheimer's disease, but for the most part, the ICs across the NIH were flat. And unfortunately, as much as our programs are interior funded programs, the Superfund programs, have been really lauded by Congress. They love the programs, but unfortunately this year we saw about a 4% decrease in those programs. So that's not good. I'll be actually in front of our appropriators, I think it's next week or the week after, and hopefully trying to make the case that these great programs that they've been very supportive of are worthy of some additional funding. So that's where we were for FY24. Next slide, please. Now, the good news is that President Biden has been pretty supportive of the NIH. And so for his proposed FY25 budget, we're looking, he's proposing a 0.3% increase, certainly nothing like the multi-percentage increases that we've been appreciating over the past several years, but in the current fiscal climate, any increase is a good increase. And also, he's going to try to rectify some of the decreases that we saw with the Superfund programs here. So he's proposing to actually bring bring the Superfund programs, increase them so that they're essentially flat from FY23. So I have to apologize a little bit. I'm still a little jet lagged coming from Japan a couple of days ago. So hopefully if I'm sounding a little bit discombobulated here, I can at least blame it on the biological consequences of jet lag. Next slide, please. So that was the budget stuff, but I have to just spend a minute here and just tell you about an extremely exciting event that happened just a few weeks ago. It was April 30th, when I was invited, together with many of the other IC directors, and actually Nicole and one of our new members, who I'll introduce in just a few minutes, was there with me, as well as April Bennett. So we attended an event. It was hosted at the Library of Congress, and it was hosted by the ACT, or Advancing Cures Today, for the NIH. And you can see here that there were many members of Congress there, both Republicans and Democrats. And it was an incredible event. So many of the speakers, Republicans and Democrats, went to the podium and were speaking in very positive and enthusiastic terms about supporting the work of the NIH. And some of them were a little bit apologetic that they couldn't do better for FY23. But again, Republicans and Democrats were very supportive they recognize the value that the NIH brings to the United States, and both in terms of promoting healthcare as well as promoting, the economy. So it was just an incredible event, very exciting, and it was kind of reminiscent of several years ago. I was at another meeting very similar to this, lots of support for the NIH. Back then they were talking about doubling the NIH budget. There wasn't a lot of talking about doubling the NIH budget this time around, but nevertheless, I think there was a real sense that at least there are a number of members of Congress who really recognize the importance of making the investment to the NIH. Next slide, and I'm actually oops, there we go, back here. So I actually had an opportunity at that event, after the formal presentations were made, to schmooze with some of the participants. And actually, all the way on the left is, I don't know if you know Congresswoman Rosa DeLauro, but she is a force of nature. It was just, and when she got to the podium, just everyone listened, and we're always very curious to know what she had to say. And she is very, very supportive. So I had a chance to talk about the work of NAEHS and the importance of the exposome and the importance of studying environmental exposures as it relates to health. So she listened pretty carefully. So we'll have to see. We'll hopefully have a chance to follow up with her. And again, thanks to April Bennett, who's there with me to help to arrange some of these one-on-ones. Also, I had a chance to meet with a Republican Congressman, Andy Barr from Kentucky. And that was a very engaging conversation. I mean, he's very interested in knowing about the work that we're doing at the NIH and at NAEHS. And, it was just very encouraging to hear that there is this type of support for the NIH. Also met with Jed Monocharan, who is the chairman and the founder of the ACT for the NIH. I also met with Senator Baldwin. I don't have a picture of that, but Senator Baldwin is from the state of Wisconsin. I grew up in Wisconsin. And she is really smart and very engaging and really understands the power of the NIH. And it turns out that her grandfather actually taught me biochemistry at the University of Wisconsin. So we had a little something to chat about. But it was just an incredibly exciting and energizing event. At times it's easy to get a little bit depressed about what's happening in Washington, but it was just encouraging to know that there are many different parts of Congress that really have their act together and really understand where their priorities should be. Let's go on to the next slide. So I've talked a lot about the strategic planning, the emerging strategic plan, and the 6 different areas of scientific focus. I'm not going to go into any of the details of that. The plan is, we are polishing that up. Hopefully plan to get that on the street here in the next week or two so that all of you will have a chance to comment on that. And once we've received your comments, we'll get this back out. And hopefully by the end of the summer, we'll have this published. And I'm hoping that for our September meeting, our new NIH director, Dr. Bertinelli, will be joining us, and we can be talking about the new strategic plan that will help guide us for the next 5 years. So today what I thought I would do is just update you on some of the activities that have been happening since February in some of these areas of scientific focus. So let's go to the next slide. So let me start off with environmental justice and health disparities. So next slide. So let's go to the next slide. So I mentioned this last February, and I think it was last year at about this time, Secretary Becerra reached out and actually wanted all of the operatives all of the operating divisions within HHS to do something very specifically in the area of environmental justice. So he actually charged the NIH to be doing something, coming up with 3 strategic and transformative EJ actions. And he wanted these to be done in the next 18 months. So we actually responded to that request from the Secretary. Next slide. And as I explained to you last time, that actually it was a real credit to us that both the director, well, the acting director at the time, Dr. Tabak, and the acting deputy director at the time, Dr. Schwetz, recognized the leadership that NIEHS brings to this, and they actually asked Dr. Archer, the deputy director here at NIEHS, to take the lead to develop an NIH-wide strategy, to develop some things that we can be doing in the next 18 months that will have a very specific impact on environmental justice. So Trevor stepped to the plate very quickly. I think he had a couple of weeks to actually assemble a team and come up with some specific recommendations. And you can see here there are a number of staff here, both at NIEHS and across the NIH. And they put together an NIH-wide working group with representation from many of the institutes and centers across the NIH. And next slide, and just to remind all of you that they came up with 3 different strategic and transformative environmental justice actions. And the first of which was to really build on our existing environmental health disparity centers and to add an environmental justice component to these existing centers. So currently, the health disparity centers are funded by NIEHS, NIMHD, and NICHD. It's about multidisciplinary research capacity building and in community engagement. And again, the idea here is to build on the health disparities, but also add a very strong component of environmental justice. The second proposal was to develop the Environmental Justice Scholars Program. Now, this is modeled on the Climate Change and Health Scholars Program that you've heard me discuss several times in my presentations in the past. And it's really about bringing people from outside the NIH into the NIH and helping to shape the programs that we develop around environmental justice. And the third area, transformative and strategic area of focus, is environmental justice training. And it's really building on the outstanding capabilities that we have currently within our worker training program and getting out to communities, to workers, to researchers, healthcare and public health professionals, policymakers, to build community capacity around environmental justice. So I'm pleased, next slide, to say that we're actually making progress on all three of these. So with respect to the Health Disparities Center, there was an RFI out, the responses are in. NIMHD is taking the lead on the Health Disparities and Environmental Justice Centers. So they've gotten approval from their council back in February. And again, the concept is approved with an environmental justice focus. There is a notice to intent to publish, and they're estimating that this publication will occur by July 30th of 2024. So currently, Trevor and the working group have been working to engage other institutes and centers. So NIMHD, NIEHS, we will continue with the same level of funding that we did last year. NICHD has stepped to the plate, Deafness and Communication Disorders, the NCI. And NICHD is interested. They're still trying to work through a tough fiscal climate. But we're hoping that they will be in a position to help to contribute. So hopefully if we can pass the tin cup and raise some additional money, we can fund an additional center. And if we can actually even collect more money, we can fund some additional centers. But it's a great program. And bringing that environmental justice focus, an additional environmental justice focus to these centers will be, I think, terrific. Next slide. So in the second area, the Environmental Justice Scholars Program, there was an informational webinar that happened the middle of last month. And so our staff here really was reaching out, and they're asking other institutes and centers to develop an interest statement. What is it that you, or why are you interested in joining this program? And we've also asked them to identify a liaison within their institute who will participate in the interview and the selection process of scholars that will be happening. So we anticipate that the announcement will be coming out in late of July of 2024, closing in September of 2024. So if you're out in the community and you have someone you want to nominate as a environmental justice scholar, please pay attention to the funding announcements that will be coming out. So this is an area where there's been a lot of interest across the NIH. So you can see here it's not just NIEHS, but NIMH, NIA, NIAID, well, actually several other institutes and centers who have expressed an interest. They're dropping a few coins in that tin cup as we're passing that around. So next slide. So in that third area, the community training program overview, we have now NIA is working, NIEHS, of course, is contributing to this. NCCIH, very interested in ODP, very interested in helping to fund this. So what's happened since February is that they've developed a pilot program to promote environmental justice dialogue. And very specifically, there's been work to partner with the We Act Environmental Justice Leadership Forum, which is happening in Memphis, Tennessee, in September 10th to the 14th in 2024. So they're preparing training materials and learning objectives for community dialogue. How do we get out in front of communities and help them to understand the issues around environmental justice? And actually what I'm finding is that even across the NIH, there's a lot of confusion about what is environmental justice. How is it different from social determinants of health? So developing those training materials with a very clear definition of what does it mean, what does environmental justice mean, and what are some of the objectives, things we want to be achieving through these programs. We just need to make that clear. We're also establishing partnerships. So I understand that from Trevor was telling me that we now have we're forming a partnership with the Smithsonian Center for Environmental Justice at Anacostia Community Museum. Next slide. And we're also just a tremendous amount of focus on community-based participatory research. I mean, this is critically important when we're working in the area of environmental justice. It's also very much about implementation science, a cultural competency in environmental justice training. So we've also identified within the NIH several different partners. Many of you may remember the SEAL program, the Community Engaged Alliance, that was very active for community engagement as part of the COVID-19 pandemic response. Also, they've identified partners with the Alliance for Community Engagement for Climate Change and Health, or something we're calling the ACE-CH. And of course, the Community Partnerships to Advance Science for Society, the COMPASS program at the NIH. So the EJ training, again, as you can see here, is NIA, NIEHS, NCCIH, and ODP. So we're making a lot of progress on this program. Next slide. So next slide, let's talk a little bit about community engagement. So one thing that's been clear is that one of the important things I can be doing, and senior leadership here at NIEHS can be doing, is getting out in front of the communities. We have to get out and we have to listen to what their concerns are. And to being out in front of these groups and visiting in these communities gives me a level of appreciation for the dire straits and the dire conditions that these people are living under. So with that in mind, actually I made it a point to visit the Central Valley of California. I did this just a few weeks ago. This is a highly diverse region. This is an area with many migrant farm workers. I mean, these are the individuals who are tending vast acreage of farmland, raising the crops, the vegetables, and the fruits and other things that we all, the rest of us, enjoy. But there are incredible health disparities and environmental justice issues in these communities. So this actually is an area where it was really the birth of the environmental justice and the farm worker rights movements. And these are some of the communities here, are some of the poorest and the most polluted regions in the nation. So it's truly remarkable. Over decades of use of pesticides, raising those crops that we're all appreciating. I mean, the soil is contaminated, the water is contaminated, and the communities, the people who are living there, their children are playing in the front yard, but they're being exposed to the clouds of pesticides that are being sprayed on the fields. So it's really just an incredible environmental justice issue. So next slide. And the visit there was being partnered with the center at the University of California at Davis. So there were 3 different communities in the Central Valley that we visited: the Contura Creek, Lennar, and South Central Fresno. And so I don't have time to go into all the details here, but many of you are probably familiar with the California Enviro Screen, which gives us a tangible measure of the issues around environmental justice. And you can see that these are communities where there's huge pollution burdens, high use of pesticides, groundwater threats. I mean, in fact, in one case, we met with the community and a woman shows a mason jar and the water is brown tinged. And this is the water that they're being told is safe to drink and they ought to be drinking this. It's just horrible because listening to these people, it's so desperate. They have to work the fields because they have to eat. They don't have a lot of choices. But it's terrible because they realize that they're exposing themselves and they're exposing their children to these cocktail of decades of pesticides contaminating their water and their soil. And they know they're going to be developing cancer, and they know they're going to be developing health threats, but they don't feel that they have much other choice. In addition to the toxicity associated with chemical pollution, air pollution, is also a great concern around the politics. These are people who they don't go to the polls anymore because they don't feel that will change anything. So they were asking me, Rick, what can we be doing to get more people to the polls and to actually be electing people who will then take their interests at heart to help to make change happen? So it's a sense of they're working the fields, they have to eat, but they just don't feel that there's a lot of positivity. So it's just a huge, I mean, health disparities. There are no health clinics, and there are no physicians to write prescriptions for the types of inhalers that children need, because the chemical exposures are exacerbating their asthma. So it's a terrible situation, and you can hear about all these things, but being in front of these groups and listening to their desperate pleas to do something is incredible. There was a farm worker there who actually drives the tractors in the fields. He's given no training at all to understand how to protect himself from the pesticides that he's spraying, behind his tractor. I mean, it's just irresponsible. And he knows his groundwater at his home is contaminated. I mean, he can taste the pesticides in his groundwater. But he doesn't have a choice. I mean, that's the only water he has. And so he'd love to, someone suggested, well, let's drill another well. Well, I don't have money to drill another well. So it's desperate. And it's not just one or two people. These are whole communities that are living under these conditions. So next slide. I mean, what are the things we did? And I really have to I applaud the University of California, Davis Center for working with us on this. The NIH can get there, and I'm happy to get out in front of these groups and talk about and listen to what their concerns are. I mean, the NIH does research. We can actually research these populations and develop the data to connect these horrible conditions with health consequences. But in many instances, they don't need more research. They want their environments cleaned up. They want a mechanism to drill another well. So what we did this time around, and we've been doing this more and more often, is that I'm insistent that we do an all-of-government approach. I'm happy to get out in front of these groups, as has Linda Birnbaum in the past and Ken Olden in the past, but it shouldn't be just about NIH. So I want the local, state, and federal partners to be there with me, to be listening to the concerns of these communities. Because it's the, say, California EPA or the national EPA that has mechanisms to actually fund and to make the environment better, to clean up the environment. So we actually did that. So we had members of there are pesticide offices within California. There are members of the California EPA, the national EPA, they were all there with me and listening to the community concerns and then speaking up. And what was clear is that you have all these different local, state, and federal government agencies that are working to make things happen, but everyone's working in parallel with each other and they're not intersecting their activities. So the net effect is that these poor people living under desperate conditions are continuing to live under desperate conditions. So we decided that we need to change the paradigm. So doing an all-government approach is that we're gonna commit to every 6 months, we're gonna have a dialogue with these communities. And I wanna know what the California EPA is doing to actually correct these problems. And I'd love, Administrator Regan at the EPA, they got $3 billion dealing with environmental justice issues. How are they going to deploy some of that money to actually bring it into these communities in Central California that are so desperate and are in need of their support? So we'll follow up with this, and we're doing the same thing. I think I mentioned last time about the community in Mebane, North Carolina. We use the all-government approach, and things are being cleaned up now. So we got to take a different approach. All-government approach, I think, is the way that we're actually going to make progress. So next slide. And actually, if it wasn't desperate enough, visiting with these communities in Central California, we actually went to Southern California into the Imperial Valley. And this was actually in collaboration with the USC Environmental Health Sciences Center. And this is a rural border region with Mexico. 1 in 3 children live in poverty. Very high unemployment rate. The air quality is terrible. I mean, it's excessive particulate matter in the air. And there's a staggering rate of asthma morbidity. So 1 in 5 children are diagnosed with asthma. So what's going on here? Next slide. So many of you may know that just a few years ago, the Salton Sea was the ultimate place for many wealthy Californians to go and to enjoy the weekends, take a picnic lunch, take their boats out and sail on the boat on the Salton Sea. It was a great place to go out and have fun. Problem is that with climate change and a whole variety of different variables now contributing to this, the Salton Sea is drying up. The water is disappearing. And as a result of this, all of those pesticides, the cocktails of pesticides that have been sprayed on the communities over the decades, they've made it down to the base of the sea, and now the water is gone. So the wind stirs up dust, and this is toxic dust that then people are breathing. So the wind blows, and the playa dust, it moves into the local communities, and especially children. It aggravates their asthma. And next slide. So you can see here that, I don't know if you can see on the left-hand side here, but that's a boat dock. So this is the Salton Sea, okay? And it's just, there's parts we don't have a picture of, but there's a little picnic area there with a picnic table and a boat dock next to it. The problem is there's no water anymore. So it's just all dried up, and actually the day we were there, it turns out, fortunately for our sake, the wind was calm, so it wasn't blowing up the dust. We weren't breathing this stuff in. But it's a horrendous situation. Next slide. And it turns out I actually learned a lot about the geography of California. I didn't realize that there are, all the way on the right here, it looks like there are sections of Iceland where you have percolating, the geothermal vents coming up. And there are industries actually tapping into some of the geothermal energy. And it turns out that there's also lots of lithium. And I think we all realize that lithium is really a highly valued mineral now. And so there's a lot of interest in developing strategies to mine the lithium so that we have plenty of lithium for all those electric vehicles that are the batteries for electric vehicles. The problem is that this is happening, I think next slide, it's happening, the development of these technologies for tapping into the geothermal energy and lithium extraction, it's not factoring the needs of the community into the equation. So they're going in, there's great needs for geothermal energy, great needs for lithium, and no one's really thinking about how is this going to be affecting the local population. So it's pretty desperate. Next slide. In fact, we also, as part of the tour, visited the local schools. And so that was just a firsthand indication of how this is affecting school children. Kids are missing school. It's exacerbating their asthma. And again, there's no healthcare system. Many of the hospitals are closing, and they don't have the funding to stay open. There aren't physicians to write the prescriptions so that the children who need the drugs to treat their asthma, it's not there. So it's really a desperate situation. Next slide. And so as part of these community forums, we also get in front of the communities. We want to hear what they have to say. And so it's a real earful about hearing about the desperate situations. But it's also about, there's hesitancy about these scientists coming in. There was a person, one of the key members of the community, sitting in the front row. After my presentation, he kind of raised his hand. He said, all this stuff happening, the science at the NIH, he said, I don't believe it. He said, in fact, I'm alive today because my physician was bold enough to actually not use all this stuff that you're hearing from Anthony Fauci, but he used some of the alternative treatments and he kept me alive. Because if I had used all those treatments from the NIH, I'd be dead right now. So this is a very challenging environment to come in. But this is coming from the community, and these are some of the community leaders who are working with community. So it's a combination of things. It's toxic dust, toxic water, toxic soil, toxic politics, toxic leadership. So it's a complicated situation. And we're committed, though. We're going to continue to get in there with our environmental justice programs, listening to what the communities need and doing what we can using an all-of-government approach to hopefully bring some relief to these communities that are in desperate need. And I just wish that, I wish this were just the case in Central California. But if you look across, tribal communities, you think this is bad, visit some of the tribal communities. I actually had an opportunity when I was working on some of the COVID testing and other things to actually get out and to experience some of the tribal communities. And these people are living in the lap of luxury compared to some of the tribal communities and the poisons and the environmental threats to their well-being. So we've got a lot of work to be doing. I'm just pleased that environmental justice is coming into focus, and that we as a government, in using a government-wide approach, can begin to take on some of these issues with environmental justice. So lots of work to be done. Next slide. So I talked a lot about the exposome. There's a lot of work that's going on to kind of operationalize the exposome. It's all about, as Chris Wilde originally described in 2005, if we truly want to understand the environment, we have to get beyond studying one exposure at a time. So there's been a lot of work to operationalize the exposome. I talked last time about the Banbury Conference. That was a step in the direction to begin to bring the community together to define what we can be doing so that we can be, what type of data do we collect if you're doing a series of studies with using an exposomics framework? How do you share your data? We talked a lot about that this morning. I mean, it can't just be everyone doing whatever they want to do, which is the traditional way that biologists have done it. We have to be smart about this. I mean, let's learn from the Genome Project, where initially the Genome Project was everyone doing their own thing and competing with each other, and they weren't getting very far to achieve the ultimate goals of the Genome Project. It was once, actually a real credit to Francis Collins, bringing the groups together, developing standards for data sharing, data collection. That once that happened, then the ultimate goal could be achieved. So let's do the same thing with the exposome. Next slide. Now, one of the key things that's happened since February is that the International Human Exposome Network, or IHEND project, had a kickoff meeting. And this happened in February 27th and 28th. It happened in Brussels. We attended this, actually Yusha Shui. I don't know if David, if you were there too, but we attended that by Zoom. So this is an effort primarily across a number of different countries in attendance. You can see them here. The U.S. was a newcomer to this effort. This is now a network that's funded for 3 years, and the aim of this is to establish an international human exposome network with the overall purpose of improving global research and cooperation on the exposome. So next slide. So lots of details here. So discussions that happened, there were 5 different objectives. So design a global exposome network. So the second is to identify and make available exposome tools and resources at a global scale. So evaluate the proposed network and its toolbox via a series of case studies. So can we develop those case studies where it all becomes real? How do we apply an operational definition of collecting data and how to share data to specific case studies? And develop a roadmap for future exposome research and engage stakeholders in the IHED. So I mentioned last time that we will be funding a center within the United States for the purpose of helping to define how to operationalize the exposome. And so we're expecting that center, once it's funded, will be working in a very synergistic way with the IHEN, representing interests within the United States, so we can truly be working on a global scale, collecting data in a synergistic way and seamlessly sharing our data across the global community. And that's what it's going to take if we truly want to understand the totality of exposures over the life course. So it's lots of work to be done. Next slide. Now, the exposome hasn't been sitting still. There have been quite a number of meetings with the exposome theme. Actually, Gary and I were at a meeting. I think we were the only 2 there in the room, but we were there in Japan just a few days ago. And this was the exposome and age, or aging in the exposome. So it was another one of these meetings. that were organized by Mount Sinai. And it was a very informative meeting. I learned a lot. And actually, there are a number of different strategies on how do you collect the exposome, how do you collect data around, say, developmental exposures. We heard about new strategies. I think many of you know about the children's teeth and how the teeth develop like rings in a tree. And if you use a laser and raster through the different layers on a child's tooth, you can actually get a pretty good estimate of what their exposures were to heavy metals and other chemical toxicants during fetal development. Turns out you can do the same thing with hair. We learned about some of the technologies that were available for doing that. So it was very interesting to learn about these things. I also learned about the social exposome. And actually, Daryl talked about the population, what was your this morning? The population health exposome? Or a public health exposome. Yep, sorry, the jet lag is but there are lots of different exposomes. And so what we did after this meeting is we had a kind of a breakout group. So most of us who were at the meeting actually sat down and talked about the next steps in operationalizing the exposome. What are the types of things that we can be doing? We have some of the experts at this meeting. Let's talk about this. So we talked about what can we be doing? Can we model the exposome on the Genome Project, where the Genome Project had this powerful project? You want to sequence the entire human genome, start to finish, and then you had a defined project where you had something we could start and you could finish it. So is the exposome something, the Human Exposome Project, is this something that we have a defined starting point and a defined ending point? Or is it more about developing a framework of how we collect data that goes beyond one exposure at a time? So we talked about these things. We also talked about the importance of collecting GIS data. So knowing where someone lives or lived or where they worked or worked in the past, can give you valuable insights into what their environmental exposures were. And by exposome, also we emphasize the exposome is not just physical chemical exposures, the types of things you can measure with mass spectrometry. It also includes the social, the determinants of health. What about diet? What about exercise, sleep? The quality of the community, psychosocial stress. It has to involve all of these different things. So, and furthermore, we have to be thinking if there are all these different definitions of the exposome. In fact, I was at Indiana University a few weeks ago, and I learned about the sociome. And when you describe the sociome, it sounds a lot like the exposome, but it's from the standpoint of social scientists really recognizing there are psychosocial impacts on human health. And they recognize, there's this physical chemical stuff. We don't really quite know how to do that, but that should be part of the sociome. So at some point, I'm hoping that maybe we can all come together and come to an agreement on, what is the exposome. So that's a lot of work that could be done. Next slide. Okay, let me also talk very briefly about this area of focus of mechanistic and translational biology and toxicology. As I explained last time, it goes beyond just taking outbred rats or mice and injecting them with different chemicals and counting, similar to the pathology we've just seen, counting lumps and bumps and deaths, but really trying to get in. Actually, we heard a fabulous talk today about really understanding the mechanisms of toxicology. What are the genes? What are the molecules that are being disrupted as a consequence of the exposure to these different chemicals? And it could be, going beyond just chemical exposures, psychosocial stress. What are the impacts of different dietary elements? So really getting at that. So next slide. And one thing I mentioned last time is that the NIH has really stepped to the plate and has funding now to the level of $30 to $40 million per year for 10 years, a new common fund project called Complementary. And this is a project that, again, you can see the goals here. I talked about this last time. But it's really all about developing the types of in vitro, in silico, and in chemical tools that can help us to better understand toxicity profiles or, say, efficacy of different drugs and other sorts of things. So it's named complementary. It's because it's not meant to say that there's no longer any purpose to use animals. I think there's a recognition that there are certain experiments you're going to need to use animals. But the complementary is developing, say, those in vitro systems using iPS cells, using from human-based iPS cells, where you can begin to study, human biology using these in vitro systems. So, so the next slide. So specific progress since last time is that there was a prize competition for launching the complementary program. It was $1 million. And the idea was to just throw out this competition, and people could respond, and hopefully with different ideas that can help to trigger the thinking about innovative new approaches for developing in vitro, in silico, and in chemical tools for these new approach methodologies. So the good news is that the prizes have now been awarded. It went to 20 different prizes. Each will be awarded $50,000. And there's a website if you're interested. I realize I didn't put this on my slide, but actually if you're interested, take your phone out right now and you can see the call up the website yourself. So anyway, next slide. So let me go on now with computational biology and data science. Next slide. So this is pretty quick here. So you've heard, and I've heard, that the future is really in the cloud. So it's no longer about just developing your own data repositories at individual universities and institutions, but we need to be sharing data seamlessly within the cloud. So next slide. So to help to make this happen, our Office of Data Science Strategy with Susan Gregorick and the Center for Information Technology, Sean Moody is the new director of CIT at the NIH. They've decided to create some incentives. So if you're not already using the cloud and you to appreciate what you can do in the cloud, they're providing $500 of credits to hopefully encourage people to develop a cloud account, which will give you access to tailored cloud trainings and will give you the opportunity for practice and learning for 90 days. So if you're interested, you can actually scan. I should have told you to bring your cell phones ahead of time. If you're interested, though, and just let me, send me a note and I can get you the information. So next slide. And there's some incredibly valuable learning modes that are available to you. And so lots of different places here. I don't know if David Fargo or someone from our IT group is here, but if you have questions about this during the Q&A, you can answer questions. Next slide. Okay, let's see here. Let me spend a little bit of time talking about our new NIH director. I spoke about her last time. I'm very enthusiastic about the leadership that she's bringing. And I think I mentioned to you all last time that 1 week after she became the director of the NIH, she invited the Institute and Center directors to meet with her one-on-one. And so I still remember walking into her office, her face brightening up and saying, "Tell me about the exposo." So she's very interested in learning more about the environment and how the environment impacts human health. But she also explains, I think many of you may have heard this, she grew up on a ranch it's in Wyoming. I got it right this time. Okay. She grew up in a ranch in Wyoming, and it was 18 miles from the closest paved highway. So she's really out in rural Wyoming. And I think she recognizes that there are health disparities. I mean, the healthcare systems next slide. So the healthcare systems for primary care settings, I mean, if you're living in Raleigh, Durham, in this area, we have fabulous medical facilities at the University of North Carolina, Duke, at Wake Forest. But if you're living on a ranch 18 miles from the closest paved streets in Wyoming, you don't have access to the same high-quality healthcare in primary care settings. So one of her primary areas of focus. I mean, she's very supportive of the environment, very supportive of lots of different things that happen, but she wants to see an improvement in the quality of healthcare in primary care settings. So this is all about facilitating and accelerating research advances for adoption and implementation in everyday clinical care, all for the purpose of improving health outcomes and advancing health equity for all Americans, including those living in, say, rural communities or disadvantaged communities. say, some of those communities that I referred to in Central California, in the Central Valley in California. They deserve better. And this is, I think, one way that she thinks that we can help to do this. Next slide. So she's really proposing a common fund project to support clinical research in primary care settings. So it's a primary care focused clinical research infrastructure. So she's thinking, why don't we bring some of the research capabilities that we have at the NIH and put them into a primary care setting? So bring some of that excellence of the work that we do and put it into places that could benefit from having access to that research excellence. So it's about integrating innovative research with routine clinical care, especially in some of those underserved communities. And so she's hoping to set a foundation for sustained engagement with communities underrepresented in clinical research, minority groups, rural, elderly, children, and communities where there were challenging social determinants of health characteristics. Next slide. So the approach is to essentially take the current protocols that we have and then and get them into primary care settings as quickly as possible. So enroll efforts of existing NIH studies. She actually put out a poll to all institutes and centers. What are some of the current clinical studies you have, and which are the ones do you think could be applicable, could be moved into a primary care setting? So partner with the clinical research networks where they exist, engage primary care sites and community networks, and implement study designs across the landscape of clinical trial innovations. So next slide. So what's happened over the course of the last several months is really obtaining perspectives from external partners. They're doing a lot of talking, doing a lot of listening, especially listening in public sessions. So that's all happening now. And so there will be a quick launch in 2024. The OD is going to put a modest amount of money, but $5 million in 2024 to expand existing studies, to engage underrepresented populations, and to enhance accrual and collaboration. In 2025 and beyond, using some of the money in the Common Fund budget in the Office of the Director to help ramp this up, getting more quality research into primary care settings. Next slide. So then let me just finish up by talking about some of staffing updates. So next slide. So Nicole Garbarini is right back in the room. So Nicole is joined us just a few weeks ago now. It wasn't that long, well, maybe a few months ago. But she's been working together in the Scope office together with Gwen Coleman. Gwen, great hire. She has been just terrific. In fact, as I mentioned, Nicole was with me at that meeting with Congress at the National Library National Library of, where was it? National, whatever it was, sorry. But it's just been terrific. So she's working with us now to really polish up the strategic plan. Sheila Newton retired at the end of January, so Nicole is drinking from a fire hose and kind of picking up where Sheila left off, and she'll be helping to bring that strategic plan together. So she's had a lot of experience at the NIH in the immediate office of the director. Dr. Tabak spoke very highly of her, and she's also most recently coming from she's had experience in the extramural research leading the grants information branch. But she recently came to us from NCATS in the policy branch. So it's just been delightful having her here. Next slide. And finally, unfortunately, I have to announce, many of you know George Lucier. He passed away on February 8th of 2024. So many of you know, he was a real trailblazer in environmental health research. For 31 years, he served as the NIEHS Director of Environmental Toxicology Program. It was in DIR, and the Associate Director of the National Toxicology Program from 1993 to 2000. So he was the co-editor-in-chief of Environmental Health Perspectives. So if you didn't know about his passing, I thought I would pass that on to you. It's very sad, a real prominent luminary in environmental health science. So I think that's it. So next slide. It's very important. I want to acknowledge the dedication of three of our council members. Trevor, Irva, and Karen are now moving on. They retired from our council, so they know, as you know, they're not here. That's because our last meeting was their last meeting. But we really thank them for their engagement over the past several years. It's just been delightful working with them. Now, there are several people, there are 4 members of our current council that will be retiring after the September meeting. But since the September meeting is virtual, I thought I would take the opportunity to actually hand them a certificate of appreciation while we're all together today. So why don't we start off with Jani Ingram. So if you want to come up, we have a photographer here, and I can give you your certificate. And let's see, where do we want to do this? By the flags or?
Probably not so close to the screen, maybe out here.
Okay.
Like the screen.
There we go. Here you go. It's just been great having you on. Thank you so much.
I'll make sure.
Careful, don't back up any further.
Yeah, I was gonna say, I'm getting the.
Okay, where would you like us to go? Okay.
It's too much of a distraction. So let me, that's much better. There we go, that'll do it, yes.
Great. Well, thank you so much. Great. Okay. Number 2. Who's next? And the winner is Andre. Right here?
Yeah.
Sir? There you go.
Alright, let's see.
Great. Excellent. Thank you very much for your service. Okay, and let's see the next. Let's see. Whoops. Hold on one second. Here we are. Thomas Leviste. There you go. What a rapid fire. Wow. Thank you very much. Really appreciate it. And last but not least, Gary, you want to come up and get your certificate? Yes, sir. Great. Thanks very much.
And just to also announce that Phil Bourne will also be retiring off of council after the September meeting, so
Okay.
we'll mail him his certificate.
He's not here, so we will mail him her certificate. Yep. Okay, well, that's it. So are there any questions? Yes.
I just have one comment with your Imperial Valley. So on the other side of the border, Arizona, that's the lettuce capital of the world, Yuma County, where I guess we probably are eating. If we had salad today, we had lettuce from that area, but it's highly agricultural. 2 tribes live there. It's a mess. And then on the other side of the border, because Mexico, California, Arizona are all right in that corner, there's Mexican cities that burn their trash, so that wafts over. So I mean, it's there's
Desperate.
a lot of, yeah, it is. It's really difficult down in that area. And you have migrant workers that go back and forth, and so their health care is horrendous. So
I mean, it's just every element of it is just desperate and it's horrendous. I mean, how can we let this happen in a country like the United States? And the really desperate thing about it is that they're raising all the vegetables, the healthy things that we've had for lunch, the things that we are appreciating that are maintaining our health, and it's destroying their health. So it's a huge injustice. Other comments? Yes.
Yeah, I wanted to comment, actually to congratulate you on your all-government approach. I think it is a great idea, and certainly you going there in person and bringing all of the, the partners there, I'm sure it helped. My question for you is, because I couldn't see all of this, but was like the industry or like the local employment places involved in the discussions, and how was coming from public health perspectives, I know that the public health departments are also struggling when it comes to limited resources and what they can provide. So they're certainly bringing everyone to contribute with their piece is great. But two questions, what was the involvement and the reaction from the employers, and second, how can you keep this sustainable, since like you probably cannot go to every single place, to keep the all government approach going?
Yeah. I missed the part, so there was a part that you were asking me.
Oh, so two questions:
Yep.
the involvement of the employers, and how they
Right.
were involved and reacted, and the second one. So you gave two examples of all government approach where you attended, but how is it happening? Because it seems that this is something that NIH is going to be, heavily like pushing as a successful model. Like, what is the future of this model?
Well, let me take up the first question first. It's where were the employers in all of this? And essentially the employers were not to be found. So it's we had an open forum, and actually some of the employers were there. One of the person who had the comment about they don't really believe in all this science stuff coming from the NIH. So and they hold a lot of power, and a lot of the employers have a great deal of influence on who gets elected to the local government, and so it's desperate. So I don't know what the solution is on that, other than to really try to convince the communities that there is a future, and that if there are things that they can be doing to help make change happen that will improve the quality of their health. And I think part of it is going to be using an all-of-government approach. So the NIH, we have a modest budget, and we're not the biggest IC, we're not the smallest IC, but we have lots of different obligations. So we have a modest amount of money that we can spend on environmental justice related issues. But the way I see this is that we ought to be partnering with the EPA. We ought to be partnering with CDC, with other agencies of the federal government with mutual interests. And we can all bring complementary capabilities to the table. And so that's what we need to be doing. So that's why when we talk about my community engagements, I'm insisting now that it's not just Rick in front of these groups. I want an all-of-government approach. And I want other partners to be listening to the concerns. And then having a discussion at these meetings about how can we be better synergizing? What are the points of intersection? I mean, as I explained last February, we had this community forum in Mebane, and we actually learned that the Army Corps of Engineers actually had been studying this project for decades, and they had actually plans. I mean, they knew all about the contamination. And we actually learned that the local government had a plan for what does the site look like, how once it's cleaned up, how is this going to contribute to the local economy? So developing a strategy on what can we all be doing and developing a plan going forward where we can all bring our complementary capabilities to actually make change happen within the communities. That's how it's going to happen. So, and I'd say to the best of my ability, I'm going to continue to discuss the all-of-government approaches and to engage. Hopefully this will become more of the standard going forward, rather than individual agencies feeling that they have to do it on their own. So sorry I don't have a better answer for you. Those are tough questions, but the situation is desperate enough, and it's not just one community or two communities. It's pervasive throughout the United States and actually throughout the world. And so these are things we have to do something about this. And when you factor the whole issues of climate change on top of this, especially in other parts of the world, that's even more desperate.
So I see Daryl has a question, and then
Yes.
Maria online has her hand up.
Thank you, Director Warchick. I was going to
Rick,
ask
by the way, that's good.
Okay,
Rick is just fine.
Sorry about that. Yeah, I'm formal. So I was going to ask the flip side of that question, because so with respect to the migrant workers and their employers. What about in the case of the EPA? So it's great you have this vision, and I think it's right on target with regard to the US EPA and NAEHS coming together. So the statutory authority with respect to EPA was on the last executive order, not the one yesterday morning, but the one with the J40 initiative in it, right? And so now we have EPA with authority indicating in low-income disadvantaged communities, LIDAC communities, where 40% of that money should go back to those CGES census tracts, right? And so, but what we find now, even though that's the law, where do you think the pushback is? It's at the level of the municipalities, because we know where this is, and so this is where we are. So how do you see
You've
that?
got several different levels of trying to evade the law. And actually, one other comment that I want to make is actually maybe a more direct answer to your question is community engagement. So if anything is going to happen, and maybe this is a response to your question too, Daryl, I had the opportunity to actually meet with some of the community groups. And these are individuals who are passionate. Well, they're actually knowledgeable, highly capable, and passionate about fixing things and making change happen. So the all-of-government approach can be a catalyst to help bring some of the resources of the federal government, well, resources of the local, state, and federal government to bear on things, but working together with the communities, and then community leadership, which is pretty awesome. So every community that I've been in, I mean, they have remarkable leaders who are very interested in making change happen, and working as an all-of-government approach, I think, is something that they've really embraced. So it's and just continuing to bring awareness to the desperate conditions that these people are living under. So not only doing the research, I don't know that we need to do the research when someone hands you a mason jar that's got brown tinged water on it. Maybe it is safe to drink, but I'll tell you, I'm not going to drink it. And it's outrageous to think that we're expecting that anyone else would do that. So just bringing awareness to this. So for the communities that, the farm workers who are living in these homes and their well is contaminated, they need an option for drilling another well. And it turns out that actually as part of the deliberations, there is an office in the state of California that does have funding for individuals like this to drill another well. I mean, the problem is that it's grossly underfunded. And that there's not great awareness of that resource. So this farm worker who drives the tractor, he was very pleased to learn that there's actually a place where he can turn to within the state of California to get some relief. And actually, California is probably one of the more progressive states as it relates to funding for environmental justice communities. But it's something we have to take up in this country, is we just have to bring greater awareness to the need. And we have to prioritize where do we put some of our money. Now, Daryl, does that begin to address your question? I'm trying to. Okay. Well, we're working on it, and working on it because we have people like you to actually partner with, and many of you in the audience, and Sharon Beard and many of her colleagues really working on the issues of environmental justice, worker training. I'm thinking that it's just simply outrageous that you have now a migrant farm worker who's driving a tractor, spraying the fields, and he doesn't know how to protect himself. Because there are ways of doing that. So we got to do better. Other questions?
We do have a question from Maria Savasta Kennedy.
Maria.
Hey, Rick. I was just going to say that you're really focusing on the awareness issue that you brought up, because it's not news on the local level that the tractor driver doesn't have any protective equipment and is exposed to the pesticides, as are the family members of these workers. But I do think that having somebody from the federal level shine a light brings more pressure politically to what's happening on the local level. So I think that's so important, in addition to to the all of government approach of bringing everybody together, I do think there's an important function of bringing the federal government to shine a light on what's happening and bringing attention to what's happening on the local level. So that was it for me.
Well, we'll keep doing it. We're actually hiring a new community engagement coordinator. John Shelp was someone who did a lot of that work in the past. He retired, and Erica Reed is hiring a new person. I understand she has some great candidates. So those are the people that I rely on to actually identify the communities and work out a strategy to get in front of these communities to bring bring some awareness, let them know that the federal government is there. But most importantly, it's also, I'll tell you, if you're, I can appreciate how bad it is. You read about this stuff, but being there in front of the people and talking with them, being in the community, seeing children with asthma, gasping for air because they don't have inhalers, it brings a whole different level of engagement. So we just need to do more of that. Other questions, comments? David, how are we doing on time?
We are good on time, so.
Okay. Any more comments? Anyone? Sharon, you want to say anything about environmental justice? I know you're pretty passionate about this. Come to the microphone so that everyone can hear you. Gary will share his microphone with you.
Rick, I think one of the things that we can do probably a little bit more effectively whenever I hear stories like that is that we can go out and provide training to those farm workers and to those individuals. And so we need to make the connections to find out if there are tools that we have in our toolbox here that we can bring to bear, because we have training programs through the Western Regents University Consortiums. And so that's what we're trying to connect some of these dots to make sure that we're able to do those types of things. So I will certainly be working with the crew who went out there and try to find out if there's other resources that we can bring to the table to make that happen. And I do know that we have a couple of other groups that are doing great work in that area that we're funding through our Migrant Clinicians Network, and they're training of farm workers, and they actually just did a great program out there not too long ago. And so making sure that we have access to that information and we can provide the training through some of our other existing resources, I think is good. And I think trying to figure out some of the ways that we can make sure individuals get access to some health care. I think that's one of the things that we have to do better out in those areas. And so making the connections to see if there's other vehicles and other ways that we can make that happen, I think is important. And I think one of the biggest things that we've been trying to do in worker training is making those connections with the consulates in those areas, because what we're finding is that they actually have also resources that they can bring together when people come into the country and get access to some of these resources and tools, and we didn't even know about that. So we're trying to work and trying to figure out how we can share these strategies and what we're doing with worker training, and then also some of the environmental justice groups. And so we're going to incorporate it into some of the environmental justice training that we're trying to develop for NIH and for researchers. So thanks.
I mean, all workers should be given an opportunity to learn how to protect themselves. And I have to say, I'll take this opportunity publicly to applaud you for the leadership and the worker training program. If you look at the statistics and the deliverables from our worker training program, it's truly remarkable. And every time that I brief Congress on this, they're very impressed. I just wish they would actually follow up with a little bit of extra funding, but it's a very impressive program. And yes, we don't want any more tractor drivers getting out there. And when it's a simple matter of just learning how do you protect yourself. And actually also just getting to the point too where people don't feel so, what's the term here? It's just given up on paying attention to their health. It's feeling so desperate that there's nothing that they can do. Well, there is something they can do, and that's I must say, that's where the community engagement and some of these local community leaders are really passionate. And they're the ones that we're going to be able to work with to actually make some change happen. Other comments? Okay, well, then.
So if I can have my slides, Nathan. All right. So we're hitting the home stretch. The update from the extramural division. Just 3 themes to bring to you today, but we'll have some discussion in a new area that I've decided to bring into my presentation each time, which is some just updates on policy from OER and other general things that you should know, and then we'll do a little bit of a deep dive into one particular pain point that I know we've had some struggles with our grantee community over the last several years. Just have some discussion on that. So we'll begin with some staffing updates. We've had some unfortunate staff departures since the February council meeting to bring to your awareness. So the first, Linda Bass, who has been with the federal government since 1990 and DERT since 2001 in our scientific review branch, and was most recently the deputy chief of the scientific review branch. She retired at the end of March. And Barbara Gittleman, one of our grants management specialists, retired in April. She's been with us since 2009. A real blow, Lindsay Martin, one of our young up-and-coming program officers who joined us just 4 years ago, left for an opportunity that presented itself at NIDA. So she's working as a program officer at NIDA now. And Jim Remington in our worker training program also has been with us for some time, since 2005. He retired also in March. And then just last week, Alfonso Latoni, the chief of the scientific review branch, retired after more than 20 years in federal service and has been here as the chief of our review branch since 2013. So we are in the process now of beginning a recruitment for the chief of the scientific review. But we have some new members of the DERT team, a few people in new roles. So Jennifer Baker, one that you council members will be working with closely. She's been serving as on a detail as staff assistant for about a year, and she officially took that role just after the council meeting in February. So she's working very closely with Ashley and myself and with the logistics and support of council. She brings just a wealth of administrative experience, and she has made my life so much better since she's came on with a number of ideas of how to manage the work within the division. And Valerie Bartlett is here with us in the room. So she came to us from NLM and is working as the council assistant and our guide liaison. So she handles all of the policy and procedure side of council and all of our solicitations and everything. So it's a really critical role in the division, and we're super excited to have you with us, Valerie. Very quickly coming up to speed, and I was saying earlier, we have this wonderful alignment of a brand new ECB system at the same time that we're bringing on a new council assistant to make everything work smoothly. So it's great to have you on board. A new member of the worker training program, Mia Pearson, who I think is also here with us in the room. I saw her this morning. So she came to us from the NAEHS health and safety branch as an industrial hygienist, and where she's led a lot of efforts in the protecting the NAEHS workforce, but particularly in areas of opiate exposure, working outside of NAEHS. And really, I think that aligns with a lot of the activities that the worker training program does. So we're really looking forward to, she just came back in. So Mia, welcome. my fingers flubbed there. And the newest member of the grants management branch, Irina Alva Weinstein, who actually joined NIH 1995 as a high school summer intern. So getting some very early experience. She will go on to become a grants management specialist at NIGMS before leaving federal service, working in the private sector, working with first responders in finding and navigating grants opportunities before deciding to come back to the government. And we were lucky to grab her as a nicely experienced, seasoned grant specialist that will be able to come in and do some work. So she also just joined us at the end of May, newest member of the family. Standard thing that I always do in my presentations, just updating you on things that we've been working on since the last council meeting. There's a lot, so we had to break it into 2 slides this time. So we had, just after council, we had a side-by-side meeting of our partnerships for environmental public health. And Rick mentioned the solicitation of the health disparity centers as one of the centerpieces of the NIH environmental justice. So we held the grantee meeting for the current group of health disparity centers here in this room at the end of February in conjunction with that PH meeting. It's a really fascinating 3 days of community engaged research and highlighting some of the really groundbreaking work that's being done in understanding and addressing health disparities. And I really want to thank the Kims, Kim McAllister and Kim Gray, for leading a group of our staff within the division in bringing together a science retreat. This is something that we used to do regularly and had gotten out of the habit of during the COVID years. Coming together for 2.5 days of doing a deep dive on a theme. So we chose the precision environmental health aspect of the strategic plan. And really, spent a lot of time as a division talking about what does this mean to us? How does it impact the different programs that we have? What do we have ongoing that we can leverage to advance this new, somewhat confusing area of science that we don't entirely understand, and really set the stage. So once the strategic plan is out, we're really ready to go with developing some new programs that can advance this aspect of our portfolio. Going on to just some later things. So at the beginning of April, we held a biomedical career symposium. And this is actually primarily led by the fellows within NIEHS and the intramural divisions. But this is jointly sponsored by DIR, DTT, and DERT. And just the last 2 years, we've included DERT and an opportunity to bring in some of our fellows that we're training and supporting into the biomedical career symposium, having some networking opportunities and discussing some of the challenges and pitfalls that goes along with navigating, getting started in a career in the sciences. And I think, what else do I want to highlight on this one? A lot of things going on, a number of grantee meetings. We also did a partnership, an HBCU Connect Day, where we had an opportunity to engage with some minority serving institutions and work with them on how do they identify grant opportunities, how do they navigate the system. We're largely taking the summer off. So our upcoming meetings is a little bit thinner. So we do have a workshop next week that's being jointly organized with the National Center for Complementary and Integrated Health, so NCCIH, that they're very interested in moving into the environmental health space. And so their staff and our staff, Thad Shug is the lead for NIEHS, have organized a workshop to look at what are the strategies that we can use to reduce exposures and mitigate the health effects of exposure to endocrine disrupting chemicals. So I think it's a really nice partnership for us to work with one of our sister NIH institutes to really kind of find some points of interface between our efforts and theirs. And then in September, we'll have a workshop highlighting a web tool that's been developed. We have a partnership between DERT and DTT called AWARE on autism awareness and recognition. And so we'll be doing some hands-on training around the use of the AWARE tool and facilitating some systematic review of autism and similar diseases or disorders. And then we'll have a grantee meeting for our children's translational centers in September, right around the time of council. So I'm flying through these. Policy and related updates. So this is something that Rick and I needed to coordinate our presentations a little bit. You may have heard earlier this afternoon that NIH is interested in building awareness and utilization of the cloud. And so the Cloud Lab and the activities of the NIH Strides initiative are making available to you as the research community tools and training, including $500 of cloud support to try out the cloud and see if it works for you. They're including a lot of training activities, as Rick mentioned, as well as access not only to compute, but also to Jupyter Notebooks, workflow managers to really enable cloud and explore the opportunities for cloud to advance the work that NIH does. So again, QR code if you want have your cell phone available. A number of updates from OER that I want to do just very quickly on some policy things that have come out in the last few months. These are all available on the NIH Guide to Grants and Contracts, and if you don't already subscribe to that, I strongly encourage you to sign up for the weekly email that comes out Friday afternoons, and there's almost always a section at the beginning for policy updates from OER. And you can also sign up for the open mic blog, where frequently he will release an article alongside those policy updates. So the first thing that we want to highlight is a particular pain point of unilateral closeouts. So a reminder for the grantee community that there is a requirement of getting a grant award that you, in a timely manner at the conclusion of that award, submit a closeout report, which includes a final federal financial report, a final research performance progress report, a final intervention statement, and a certification. That's required to be submitted within 120 days of the completion of the award. That includes a competitive segment. So if you submit your type 2 and get refunded on that award, you still have to do the closeout report for the previous competitive segment. There's time for review and process and working on those. At the 120-day date, NIH will then begin the process of unilateral closeout, where we'll reach out to you, say, we didn't get your reports, we need to get those, please provide them in a timely manner. If it gets to 1 year after the completion of your project, we will report noncompliance to SAM, to the System of Award Management. And there are real consequences to that. That may impact your ability to receive further federal funding if you do that. And importantly, policy is retroactively effective. So we are going back to projects that were completed as early as January 1st of 2023 to enforce the unilateral closeouts of those projects. So please pay attention to your closeout requests. Please pay attention to the grants management staff and the program staff when they reach out and say, we need to get this moving because there are potential very serious consequences to a unilateral closeout, and we want to avoid that. And for each of these updates, I put a bit.ly in, so I'm not savvy enough to do a QR code, but I simplified the URLs so that if you want to see those actual announcements, the links are there. Foreign subawards is a second pain point to discuss. And again, this is not a new policy, but this is an updated guidance and enforcement of existing federal policy. So foreign subrecipients are, so a subcontract to a foreign site. They're a standard NIH tool. We encourage them when they are appropriate and support the research that we are funding. There is a requirement that when you negotiate your subaward with that foreign subrecipient, that they provide you with access to the notebooks, the data, all documentation that supports the research that they have conducted in support of your project. They have to provide access to those materials at least once a year to align with the time that you are submitting your RIPR, your progress report. It can be an electronic access, so they don't have to send you their physical notebooks, but they do have to provide you with access to that. We will not support any subaward that does not include a term that includes you having access to the materials from the subawardee. So this is growing pains for us as well. This is new enforcement and new guidance that we've received, but we have to be compliant with the expectations from above. Good news for our trainees. The NRSA stipends were finally released between our council meetings, and this year we have a significant increase to our trainee stipend levels. So a 4% increase for our predoctoral fellowship and 8% increase for our postdoctoral fellowship stipends, and small increments to the training-related expenses and the dependent care expenses. And this is another change that I think is flying a little bit under the radar. It is no longer child care, it is now dependent care. So those funds can be used for other dependent needs, including elderly parents or other justifiable dependent care costs. This is a retroactive to all fiscal year 2024 awards, so we are revising all of the fellowship awards that have already been released, and our T32s are being awarded at the new levels as those are going out. And just a truth in advertising, an increase to the stipend levels does impact our training budget. And as with what most of the universities are seeing as they are increasing trainee stipends, they're having to reduce the number of trainees that are being supported in order to balance their budgets. So there's no assurance that the number of trainees, the number of slots on our training grants, the number of Fs that we award is going to be maintained at previous levels. I think we're going to do everything we can to get close, but we do have to balance our budget and our priorities. We are not, as of yet, considering actions that some of the other institutes have taken of capping the number of slots on an institutional training award and those kinds of things. But we do have to balance our budget. So more to come in this space. And I think the last point to raise on this is that along with the guidance, there was an expectation setting that these similar, not guaranteeing the absolute percentages, but similar increases in stipend levels are anticipated over the next 3 to 4 years. So the goal is to bring this up to a living wage for the stipends. And I think my last policy update from OER is something that we talked about before. Noni Burns, when she came last year to speak to council, talked about the efforts that CSR is doing to simplify the review frame, the simplified review framework. So while we are still bound by the same 5 criteria to judge scientific merit of significant innovation, investigator approach, and environment, we're bound by law to use those 5 criteria. They've been reshuffled into 2 fundamental questions. The first one is, should the work be funded? And secondly, if it is funded, can it be successful? So it's trying to simplify the questions to make the life of the applicant, the life of the reviewer a little bit easier. And also the investigator and the environment criteria are no longer going to be scored criteria. They're going to be acceptable or not. So the hope is that this will reduce some of the reputational bias that we know underlies NIH review. They've also moved several of the non-score driver factors of the review from the review meeting to program staff. So this places a lot of burden on our staff. So please be gentle with your program officers as they try to work through these things. But hopefully there's a hope that this will reduce the burden on reviewers and hopefully encourage more people to participate as reviewers on applications. So a reminder, this is not an announcement, but a reminder that policy takes place for all applications received January 25th of 2025 and later. And CSR has really been working hard on trying to develop all of the training materials, the new forms and everything that will be used to support those applications out. So I think the guidance for reviewers is already out on the OER webpage, again, linked with a bit.ly there. And the guidance for the applicants and the new forms should be coming out. I was hoping it would be out before council, but it is not out yet. But within the next few weeks, that we should be getting the new forms and the guidance for those. Now, deep dive on a pain point, unobligated balances, and just having some conversation of what they are and what are some of the tools that we have in our toolbox to manage them. And just to kind of set an expectation, we don't have a way of really of knowing just how much money we have in unobligated balances across our portfolio, but we estimate it somewhere in the neighborhood of $60 million, which is about the same amount of money that we put into new awards every year. So it's a significant amount of money that is sitting unused in bank accounts right now. So I want to delve into a few definitions to hopefully decrease some of the confusion around what is an unobligated balance and what is a carryover and some of the other things that we talk about here. So an unobligated balance is an amount of funds that are awarded in a previous year that are not formally associated with a specific expenditure. So you can have a plan in the budget that we're going to be supporting a postdoc, we're going to be supporting this much in our animal models or our human subject recruitment and all those things. If it's not an actual person that you have identified to hire or a specific purchase that you have already initiated, it's an obligation. And if it's money that's remaining from a previous year, it's just sitting in the bank account right now. When you submit your FFR, your federal financial report, you can include a line for an unliquidated obligation. So that's money that's still in your bank account from a previous year, but you've associated it with a very specific expenditure. So you've ordered a piece of equipment that hasn't been delivered and invoiced. That's an unliquidated obligation. The money's still in your account, but it's already been promised to a very specific contractual agreement. A carryover is not the same thing as an unobligated balance. A carryover is a request to utilize funds from a non-obligated balance to a specific need in the current grant year. So that has to be funding beyond what is already supported by your grant in that year. And we'll get into an example of how that works in a few slides. A drawdown is an expenditure from your grant account. So you have an account in the payment management system that has all of the funds that were awarded to you. And you will, as you spend, you'll be drawing those funds out of the payment management account. So that's a drawdown. And several institutions have a practice where they will pay out of a central account and then replenish that from their grant accounts as the year goes. We can't see the spending that is happening in that holding account. We can only see the drawdowns. So as we are looking at your expenditure through the year, we see unobligated balance from the past and any amount that has come out as a drawdown from your grant account. You may be thinking that you've spent lots and lots of money, but if it hasn't been pulled out of the grant account, we think you haven't spent anything. So it's really important that you work very closely with your budget office to understand what is the actual spending on your project as it goes, and communicate that back with your grant specialist and your program officer, so that as we're asking questions, you've submitted a supplement request, we see that you haven't spent anything. We need to have some certainty that you actually do that because of the last item here, the bona fide need rule. We are not legally allowed to award you more money in any one year than you can reasonably be expected to expend in that year. That includes the new award. It includes any supplements that we award to you on top of that, and it includes any amount of unobligated balance that you have sitting in your account. So if you've got 100% of your annual budget sitting in an unobligated balance, it becomes very difficult for us to justify to the auditors why we need to give you another full year's award. So hence the discussion. The last thing I want to talk about before I get into the examples and some made up graphs is SNAP, the Streamlined Non-Competing Award Process. So this is a modification of policy, of provisions in policy for progress reports, notice of awards, and financial reporting that for most grantees, that's almost all of our R's and K's, makes their life easier. They can just do things and then tell us about it later. And most germane to this conversation is for applications that are on SNAP, the FFR is only due at the end of the funding segment. So you get a 5-year R01, you have to submit the FFR at the end of year 5. You do not have to submit one every year in there. So we're not getting an update on your actual unobligated balance. We have the line on your progress report that what you estimate your unobligated balance is, but we don't get a report on your unobligated balance until the end of the project, which is part of the pain point with those drawdowns on those, as you can see. Non-SNAP awards are most things that are not R's and K's. So center grants, cooperative agreements, fellowships, including institutional training grants, small business grants, clinical trials are not eligible for SNAP. And then we have the ability to remove projects from SNAP when they chronically don't report in an appropriate manner on the actions that they are taking. So it's not a one-time something was a little late, it's when there's a pattern, a repeated pattern of a failure to report. We can remove projects from SNAP. So let's dig into how does an unobligated balance happen. So I'm going to use the same system for all of the next several slides. So in a given grant year, year 1 here, we make an award. average $500,000. Ours is about $460,000, I think, is the average R01. And that grantee goes through the year. So that's the amount awarded day one of the grant. They have all $500,000 in their bank account. They work through the year and they spend an amount, in this case $400,000, to do the work that they were funded to do, which leaves a new unobligated balance of $100,000 at the end of that year. The next column will be carryover. Again, in this case, there's not a carryover, so you'll see that in the next slide, I think. And then you have the total unobligated balance. That's what actually should be reported in your progress report, not the new unobligated balance that you generated this year, but the total amount of unobligated balance. And that number should be, under SNAP, you're able to carry over 25% of that unobligated balance into your current year without prior approval. So how do they grow? And so each set of bars here is a grant year 1, 2, 3, 4, 5 here. So this awardee consistently, every year they receive $500,000, they spend $400,000, which means in the first year, they've generated an obligated balance of $100,000. That's okay. Second year, it's up to $200,000. And by the end of a 5-year program, it's $500,000. It's another full year of funding. That's a problem. That's where we end up with a lot of money sunk into unobligated balances. So this is what a carryover then looks like. So again, same project, $500,000 a year. First year they spend $400,000, second year they spend $450,000. The third year they have a large equipment purchase that they need to make. That's going to cost more than the $500,000 that they are awarded in that year, along with supporting the work that they are doing. So in this case, it's not a large carryover. They're a SNAP grant. They can just carry over that $100,000 shortfall that they were going to have to support that. If they're a non-SNAP grant, or if it's more than 25% of their annual award, they have to submit a request to us for prior approval to be able to do that carryover. So then they take that unobligated balance from the previous year, apply that to the current grant year, and now their total unobligated balance is less than it was previously. Then they go back, they continue their spending at a reasonable level, and they end the project with a modest unobligated balance. So what are the tools in our toolbox to manage unobligated balance? So for the last couple years, we have been using what we call mid-project no-cost extension. So this is a break in the middle of your funding, where we give you a year without any new funding to spend down unobligated balance through a carryover. When we do this, we add another year at the end of the project period. So you still get your 5-year R01, and you get all of the money that you were originally committed to. So you get 100% of the funding. We don't reduce the commitment. That 5-year project just becomes a 6-year project now. A new tool in our toolbox that we've started using this year is a restructuring. So this is a redistribution of funds from the current year, so that you can use your unobligated balance, carry over unobligated balance to complete the work this year. But we increase the amount of funding for the remainder of the project to allow you to keep we don't extend the life of the project. So a 5-year R01 is still 5 years. We don't reduce the amount of total award that you get. It's just the distribution of that award between the 5 years gets changed. And then, and so our proposal is anything that has 100% or more of their grant balance as an unobligated balance is likely to be a mid-course no-cost extension. At a 70% unobligated balance, we'll begin looking at the probability of doing a restructuring of that award. And then the third tool in our toolbox is an offset. So when there is not a need for the additional funding for an unobligated balance, or a chronic pattern of underspending, we have the option of doing, rather than a restructure, an offset, where we reduce the amount of award in the current year allow you to carry over unobligated balance to complete that work, but we don't increase the out years. So you actually will receive less money over the life of the award when it is offset. We do this regularly with some of our programs, like the T32s. There's a routine offset of those. So this is an example of how a mid-course no-cost extension would work. Again, they receive a $500,000 award. The first 2 years, they only spend a total of $500,000. So at the end of the second year, they've got a full year of their funding already in an unobligated balance. So in the third year for this project, we don't issue them an award. We approve a carryover of that unobligated balance to support the work through the year. They spend $400,000, so they still have, at the end of that, $100,000 of unobligated balance remaining. Your grant number will not increment during that no-cost extension. So year 3 is still year 3 at the end of the 4th calendar year of funding. When you restart the award, we issue a new grant award, it increments to year 4, or year 3, sorry, at that point. Yes, Gary.
So in that situation of a mid-course no-cost extension, at the end of the 6th year, can they still get a no-cost extension?
They can have a second no-cost extension. It has to be approved.
That's okay.
So they can submit a request for a second no-cost extension. It has to be approved. In theory, there is a possibility of a third no-cost extension. It requires approval, I believe, at RICS level. And in my 23-year career in extramural, I have never seen a third no-cost extension justified. But it's a good question. So thank you for asking it. And that does count as your first no-cost extension. So you can't get 2 more years unless there's some truly exceptional circumstance. So in the case of a mid-course no-cost extension, the project period is extended for a 6th year, 6th calendar year, but the total amount of funding that is received is unchanged. So you still receive your $2.5 million over 6 years, not 5 years. This is an example of a restructure and how that works. So this is a case that's fairly similar to the last one. I didn't change my numbers enough as I was creating these cases. Sorry about that. But in this case, we've got an obligated balance between 70% and 100%. We're not comfortable funding the full third year out of the unobligated balance. So we issue in the third year a reduced award, in this case $150,000, and approve a carryover of $350,000 from the unobligated balance, allowing $500,000 to support a $500,000 spend in year 3. And then years 4 and 5 are no longer $500,000 awards, they're now $650,000 awards. And the investigator is able to spend at those levels. The unobligated balance remains relatively modest and within the 25% of a 1-year budget. So in this case, sorry, I didn't increment my things. So again, the third year we have a carryover, but there is also a non-competitive award issued. The grant year cycles, we don't extend the project. The future years are increased. And so the 5-year total is still the $2.5 million. It's just the distribution within that period is different. And then this is the case of an offset. So in this case, the grantee is not increasing their spending. So they still have between 70 and 100% unobligated balance at the end of the second year, but we don't have the confidence that they're going to be able to spend at that full year level. So in this case, we don't restructure adding the out year funds to the end. We instead offset the award. We do issue an award with non-competing dollars, fund with a larger percentage of the unobligated balance, and then the out years, in this case, are not increased. We don't add a 6th year of funding. The 5-year award overall is reduced from what was the original commitment of that project. Then the last topic to talk here is Related to this, it is certainly related to the challenge of unobligated balance, but it's a special case of the use of offsets that we are planning to implement as we move into fiscal year 25. And the rationale for this is that when you submit a Type 2, a competitive renewal of your project, you have requested a new budget to support the new aims of that project. The previous aims are no longer the aims of the project. The budget that was intended to support those aims is no longer needed to support those aims. So why are we carrying over an unobligated balance from one funding cycle to a new funding cycle? We've got another 5 years to do 5 years worth of work. So the conclusion is that carrying an unobligated balance through a Type 2 is not justified. So beginning in 2025, we are going to be implementing a standard practice of offsetting the second year. Again, we have to have the federal financial report from the previous competitive cycle. So this is an action that will be done on the second year of a Type 2 funding cycle, of offsetting the unobligated balance of the previous funding competitive segment, allowing a modest carryover of that 25%, as long as we are consistent with the bona fide need rule. So what this looks like, the first column here is grant year 5. So that's the last year of your previous competitive segment. You had a $450,000 award, you spent $400,000, you have added $50,000 to your unobligated balance, which is now $200,000. It's a reasonable amount of money. This is not somebody that we would have been targeting for an offset or a restructure in that previous competitive segment. The year 6, you apply for and are successful getting your Type 2 award. You asked for a modest increase to the budget, $500,000. You spent $450,000. You add $50,000 to your unobligated balance, which again carries over from the previous funding cycle. So in the second year of the new competitive segment, we look at that $200,000 that you had left at the end of year 5, and we say, okay, we're going to allow you to carry over $125,000 of that, and we're going to apply the rest of that as an offset to your year 7 award. Again, you can continue to spend at the $500,000 a year level, and you maintain that unobligated balance. And the total amount of new funding coming into that second 5-year segment is less than the $2.5 million that you requested, but the total amount of money that is supporting the research of that 5-year cycle is the $2.5 million. You're just bringing that carryover from the earlier competitive segment into that.
So I lost $100,000.
You lost $100,000. But I was able to put $100,000 into a new grant, so. Okay, I'm sure that there are questions. So I'm going to say thank you all for your attention, and I am happy to take any questions that you have on any of my content, but particularly this unobligated balance discussion.
Thank you for making that easy.
Daryl says thank you for making it easy, so I've been successful. Gary.
Kind of an observation is you start off by being apologetic for like the unilateral closeout. Just enforcing the rules.
Yeah.
I don't think you should be apologetic.
It's not apologetic, and I'm sorry we have to do this. I'm sorry that this is a pain point. It's new for us as well as it is for you.
But I also think too is that most organizations and businesses follow their budgets. You as an institute follow your budget, and I think that NIH grantees, we just got very sloppy by thinking we just have a slush fund. And I think it's totally reasonable to follow these things as a council member and a grantee as well.
As a dean for research, how do you feel?
what you were saying.
But thank you for that, Gary. I do appreciate it.
So this is Steph Cormier.
Yes,
Thank
Steph.
you very much for your discussion on that. I think that was very helpful. So the things we struggle with, I mean, while it's easy to say for P grants, with the R grants, the P grants are a little bit more difficult. The P42s in particular, right? And that first year was COVID, so we've got about $500,000 in unobligated balance that our institutions aren't allowing us to touch because we don't have carry forward approved. So how do we cross that hurdle and get the money we need so that we're not offset? Because what I just heard is you might offset our SRPs if they're renewed in the next cycle with a type 2 offset. Did I follow that right?
You
Okay.
did, in fact, Steph, yes.
Okay, so I'm scared now.
Yeah, no, I mean, there's pressure on us to process carryover requests in a timely manner as well. So this is not only pointing a finger at you; it's a recognition that we need to be responsive, and it does need to be a partnership between our staff and your staff. You and your budget office, the four of us, program, grants management, PI, and sponsor programs need to be working in coordination on all of these so that we can address these pain points. We need to make sure that we understand the challenges, the realities, but as Gary was saying, you need to also understand we have a balance that we need to budget, and if this is the doubling and we have a 35% pay line, we could have a different conversation than when times are really very, very tight.
I think y'all have been very responsive. Don't get me wrong. I actually think that it's the institutions that have their own policies, and every time we ask for clarification from NIH says, within your institutional policies. And so what it does is it throws it back into this cycle that we never leave this loop. And so if y'all could actually give us a really clear directive that we can actually spend these unobligated balances for what they were intended, that would be great. That's all I got to say. Thank you, David.
Yeah,
I appreciate it.
My comment, rather than a question, is just a follow-up on that you're speaking to a relatively small audience here of your fundees, and I don't think that anybody at our institutions is listening in to really wrap their heads around that. What are the next steps to kind of get everybody on the same page?
So the recording of this will be on YouTube in about 2 weeks, so you can tell your sponsor programs, you need to listen to that conversation.
I can't make them listen, though.
Yeah, that's true.
Yeah, no, I think we need to have better conversations, and it's that four-way conversation that needs to be had. I know from my conversations with the grantee community, the PIs, that it's pretty universal that you're all feeling the same pain point. But I've also talking with Jenny and the grants management specialists, and they're talking, and there's pain everywhere, and I think that we can work together to decrease it. Jenny.
So off of budgets, but I might have misunderstood, but when you were talking about sort of a different strategy for review or funding decisions, it sounded like that step one is, can we fund this, and step two, can the science be done? It seems like that's backwards.
So it's a single review meeting that is, rather than focusing on what is the significance of this project, what is the level of innovation as 5 different criteria, it's reframing the conversation to, is this important work to be done? And on top of that, is this important work that can be done? So it's reframing, significant innovation is that, should it be done?
Yeah.
And then the can it be done is primarily approach, but it also includes a subjective on the environment and the
Right.
investigator.
I guess just, it seems like if it can't be done, then even if it's important, it shouldn't be funded, right?
Well, so funding is our decision.
Yeah.
You know, so we always will look at this. This is really getting through the evaluation of the scientific merit. And yes, you know, the reviewers are still going to be judging an overall score. So there's still going to be an overall priority score that is assigned to the application that balances should it be done and can it be done. You know, NIH gets a lot of heat, shall we say, for funding a lot of work that can be done that is very incremental.
Yeah. Okay. I understand.
So
yeah.
I think there's some.
Yeah. I just was
think
thinking,
part of this
I'm going to
is
fund this
trying-
because it's really important, but the science is not possible. But, you know, I don't know. It just came across weird to me. So.
So I have a follow-up question because there was something interesting. So at CDC, we are also going through a simplifying our grant proposals and review process. So my question is, will when the reviewers review for these two topics, will there be like sub-questions that they will score, or will they be giving like one score for number 1 to 10? Like, how are you gonna rank
Yeah,
it?
so it's the 2 criteria score now. So under the new framework, it's the should it and can it will be scoreable elements, and then there will be an overarching priority score on top of that. There will also be a subjective acceptable, not acceptable for the investigator and the environment. And then there are other things that we will be asking the reviewers to comment on, and I do not remember off the top of my head what they are. There are some criteria that they are currently asked to comment on that they will still be asked to, and then there are other ones, I think the budget, the use of select agents, and something else that are being moved to the program staff and grants management staff to evaluate. So those will no longer be review criteria. Other questions? If not, we'll change the agenda to the open council discussion. And Rick actually teased this in more detail in his presentation. But Dr. Bertinoli asked us specifically to bring the topic of her research in primary care settings to council for a discussion of where we as the environmental health sciences, and somebody needs to mute on the Zoom, I think.
I was just sitting here.
Actually, I think I'm a host if you want me to, if you can go over and mute. No, it's on somebody on the Zoom. I think we got him now. So, you know, as Rick laid out, the health of the U.S. population is declining, and that decline is steepest in underserved and underrepresented populations. And Monica would like to establish a program to integrate research and primary care across all mission areas of the NIH. So that the thought is by integrating innovative research into routine clinical care, we can really address some of the greatest challenges that are facing communities. The proposal is for a $25 million investment, scaling up to $50 to $100 million per year within the Common Fund to do this. And we're in a process now where she would like to gather feedback on where do you as council see this impacting environmental health? Where do you think that we have specific strengths or efforts that we can contribute to this? Where will we be able to fit in and leverage with a larger NIH-wide effort to engage clinical care, primary clinical care, and clinical research together. So with that, I'm just going to leave that slide up, and I'm going to move back over so I can take notes. And Rick,
David, if I could just add one nuance here, for at least to start this out, she's looking to kind of enhance existing programs. So one question for council is, are there some existing clinical programs where we can add the environmental health perspective to these programs and offer them up as opportunities to, get these into primary care settings?
Well, the CATS programs, right, would be a good place to work with these. The problem is the LCATs are probably mostly in urban areas.
Yeah, so I think that's one of the challenges that Monica is particularly interested in getting thoughts, is how do we, get into less urban areas. And I think, one of the unique things, it's the conversation that we had earlier on environmental justice. Many times those are in urban communities, but they're underserved parts of urban communities. And so where is the opportunity to leverage environmental justice communities, whether they are rural or urban? And Janie, I see you have a question, comment.
So one thing that we're doing, it's part of an American Cancer Society grant, but it was required. But in the populations that we're studying out on Navajo, is to do like a ground rounds to both disseminate what we're learning about environmental health exposures to the healthcare workers at a hospital out on Navajo, so that, because part of the problem is the healthcare workers tend to cycle through, because there are some permanent people there, but there are also people who come for a few years and they leave for various reasons. And so trying to make them aware that there was all this legacy mining out there, and these are questions they should be asking, but just maybe having this as a piece of, like NSF has the broader impacts or something like that, where it's like you need to figure out how to do this, whether it's in a hospital setting or a clinic, because there's a lot of more clinics out in rural America as opposed to a true hospital. But, but it would have to be for, I would think, a researcher who is doing work there, right? Who understands and what the problems are, but to really try and almost force it kind of thing. I know I have this former student who just finished his MD, and he did just that. He was at 3 different clinics and got really good feedback from the docs and from the nurses and so forth about the information that he was providing. So I think it's one, it doesn't take a ton of like extra work. It's not like a clinical trial or anything like that.
So I know that, we're dealing in public health with rural population being very much underserved by healthcare. There are a lot of clinics that are closing, and their access to physicians or any healthcare facilities getting longer and at a longer distance. And I think probably this is also what you referred when you were just discussing the 2 places that you visited. They didn't even have access to having somebody prescribe medication. So during the COVID, telehealth services were something that was utilized more and more. So that has been like one way of addressing it. And I was wondering if, this has been incorporated as a possibility for doing research on one side, and on another side, the digital equity. So there is currently funding coming for digital equity to all states, and they're still in the phase where they have to think through how to utilize the money and prioritize. And I know that health services is one of those, and accessibility for telehealth. So that was my question, if there is telehealth incorporated in this proposition.
Actually, I don't know the answer to that question, but that's a really good question. I know when I was living in Maine, telehealth was a very effective means of getting healthcare to many parts of Maine that were distant from some of the major medical centers. So it's, I don't know, but that's actually a really relevant point. Gary?
Kind of a follow-up on that, it would seem that from a clinical research standpoint, I'm trying to get an idea of like exactly what is envisioned in doing that, but it'll start with a clinic visit, right? A primary care connection, but the likelihood of follow-up in person is very low. So it seems like there needs to be like an app developed so that when the person leaves, they can do the follow-up background information on an app from their home or a telemedicine sort of thing, because it's even harder to get the follow-up visits in these sorts of situations. So like if the Common Fund developed like a really nice app where you could do, then you could start thinking about, a questionnaire that addressed some environmental issues where like with that module had that environmental piece. But if you don't put it back into the citizen's hand sort of thing, I don't know how you'd have the sort of follow-up for clinical research. Like it has to go back into their hands because they might go to the doctor one time in that year.
I mean, I think that's a really relevant point. So that could be a specific recommendation we make. And so someone's taking notes of all of this, right, David? Yeah, okay, so we're recording this. Also, just the whole issue of getting an electronic health record that has information relating to environmental exposures. We actually talked about that in Sapporo. It's a desperate need for physicians to ask their patients, where do you work? I mean, are you an agricultural worker? Are you someone who's working in these fields and likely to be exposed to pesticides? And what are the things you can be doing, you can be recommending? So that's very much about primary care. But it's, I think the real opportunity for us is to get more of the environmental exposures into the whole primary care setting, because it doesn't exist for the most part now.
Yeah, so this is actually a question, and Karen, I'm going to put you on the spot because you presented that you have had some success with integrating EILER into VA clinical care. And how has that experience worked, and how have you kind of worked with the physicians to be able to understand and integrate that information into their management of the patients?
Oops. So a couple things. When the PACT Act was passed, there was one requirement that the physicians had to ask those questions. So they came up with a toxic exposure screen, and it was a very simple ask. It was like 5 questions. Have you been exposed to Agent Orange? Or are you concerned about Gulf War, or burn pits, so there were 5 simple questions on that TES, toxic exposure screening, and then that put a flag into the system that if there was a concern, I think there was 40% of veterans that said they did have concerns about toxic exposures. So then they had an exam, but if they didn't have any concerns, they would be followed up within 5 years. Regarding the physicians and sitting down with, the EILER, I don't know if they use it as much as VBA uses it right now, but, they could say, oh yeah, I see you were in this location. I can see that this, this incident was reported at that time, but we don't really have any research connected at that point. So I'm listening very carefully to see what ideas you guys come up with, because, it could be a very important aspect for us too.
So I'm curious about your reaction to the idea of integrating it into integrating innovatively into existing routine clinical care, because I think the VA is an excellent example where you have a robust countrywide network with community-based outpatient clinics, and I can see how there could be some real success there. But if you're talking about rural clinics or places that are farther from, urban city centers, you have understaffed, overworked physicians. You're going to ask them to do another thing. And so I'm just wondering if there's a feeling that maybe we need to be a little more thoughtful about how we approach those communities, what resources we bring, make sure that we have follow-up, that we have long-term commitments to those places where we can have those commitments.
I think you're exactly right. And so I think then researchers have to take on that role and not just say, well, train the trainer and then you get to do it or whatever. I know the group at University of New Mexico, who was led by Johnny Lewis, and now Deb McKenzie is there. I mean, they've really taken on that model more in terms of having their folks, and whether they're even, they fund community health workers to go in and do some of that work. And so it's also community members that are bringing that information, but not saying, okay, you as the nurse or you as whatever need to do that. The other thing I didn't mention that I think is important is I also had a sociology student come up with environmental health survey just to understand what is the baseline. What do people even know about these exposures in this area? The tricky thing is, I think, is going to have to be very specific to where you are, right? You can't, like, having the Agent Orange, and those are great questions, but that's not going to work if you're on Navajo land versus Choctaw land versus, at least as far as Native communities and other places. So that's where I think, as people are doing work in those areas, sort of strongly encouraging them to do that kind of thing and work with those health clinics or whatever it is.
Any other thoughts or comments on this or any other topic from council?
I just wanted to mention the fact that, if I could, going back to the director's upcoming initiative, right? And so there, one of the things that we would be concerned about is in terms of keeping the train on the tracks would be the historic, acknowledged underrepresentation of algorithmic bias in EHRs to begin with. So that has to be a starting point. And I think that she probably had the fact, because improving research equity, that's right there. And so that's what that part is about. And so if you're going into, doesn't matter where you go, tribal lands, inner city America, rural America, that bias is going to be there in that segment of the population. And so because these large learning models, language learning models are coming on board, and so you see if you're underrepresented initially, then you can see how those models will amplify that, the machine learning and the language learning models. So in patient condition information, which is going to be a part of the Internet of Things, right? Biometric data, DNA. See, all of that's in that cycle. And so we've got to take that into consideration as this is being thought out. And so we have a paper coming out on that, by the way.
Yeah, I think that Monica's pretty aware of that. It's just integrated. And the reference to health equity, I think, is a way of really embracing that.
Just two observations from the day, and I think they've been across the board, and you've, Rick, have emphasized them today and in the past. We've been in this space before. One is that one government approach. We don't always call it the same thing, but the PACT Act, the TIRWIG, and what we're doing, all that work across all of that NSCC committee is really about coordinating and leveraging. We don't call it one government, but I can see that that's exactly where we're going, trying to do complementary, non-duplicative, amplified work. You've got it in spades with your visit there, but I think that's something that we ought to encourage just about everywhere. So right on board with that. And the other thing that we've talked about repeatedly, and again you raised it, it's about data sharing. You know, starting early as we're formulating research. We've taken a different tack because we've seen it in all of our committees, not only on this NSDC activity. And instead of trying to define spaces where we have maybe one type of database or something, just look into the options for looking at interoperability. So we all keep our own little databases or whatever it is we're developing. I also heard, for exposomics specifically, trying to look at something, opportunities for doing a common etiology. I think that's really good. But beyond that, sort of maintaining, understanding that we can't have one database or we can't share unilaterally, but we can work towards interoperability. And that's terms that I think we're all aware of. So those two things I heard amplified from what I'm experiencing in this meeting today, and I think it's ones that we could certainly carry forward.
I mean, that seamless interoperability, I think, is really critical. And if we don't do something proactively to organize it, simply around ontologies, we're going to continue to have a bit of a mess where everyone collects it the way they want to collect it. I mean, Monica Bertinelli had a great, and sorry if I've already told you this, but I think it's a great analogy. It's the issue around data sharing is that it's kind of like toothbrushes. Everyone recognizes, you need a toothbrush, we should be doing this, but no one else wants to use anyone else's toothbrush. So that's part of the problem. But we have to, at some point, recognize that we have to do something differently because we're not benefiting from, well, there's a lot of duplication of effort. I mean, I see that across the data resources that we sponsor just at the NIH. There's a lot of duplication of effort, and we just need better synchrony so that if we collect the data in ways that make it more seamless to share it, it just makes it easier down the road. So it's a lot of work that we can be doing. But I think that's governance. We have to come to some agreements on how we do things. And I think we also have to be very careful. A lot of times we get the money and then there's this obligation. You got to get something done immediately because it's really important. Maybe we ought to be spending more time to be thoughtfully considering how do we work collaboratively together. I mean, I think some of the things, and I'll point out Gwen, who's doing a great job with the Climate Change and Health Program. It's really about getting people working in synchrony together, and it's as Gwen can tell you, that's not easy, but she's been doing a great job on this. So lots to do, and and a lot of this is about culture change.
Mm-hmm. I agree. Maybe we have the benefit for our JEEP subcommittee that we don't have any funding, so that what's allowed us to do is to come up with those strategic plans and those ideas, and we're actually starting to work on some of them without the funding, and also sort of going through the thought process, particularly on that data sharing component. So getting those ducks in a row, so to speak, first. And then following. But I couldn't agree more. That's an area, and we've talked about it for many years. And so if we could finally begin to act on it, I think that will be both of those areas, I think. I'm totally
It's up to us to do that. And we also have to get away from the model that we can't do something differently unless there's funding to do it. So maybe we ought to be thinking about what's a smart thing to be doing, and then come up with some agreements on how we can work collaboratively together. I mean, the example of the climate change and health, that all started before there was funding. So just working together because we all recognize that the challenge is much bigger than what any one person can do. So we can work on that.
So with that, I think we're at time. So I would like to thank Ashley and Valerie and Jennifer for all their incredible behind-the-scenes work to make this meeting happen. And also thank Nathan and John and Chris and the folks in the booth for making the AV work and sync with the Zoom for the participants and the webcast for the public participation. I think this has been it's been a long day, but I think it's been a really insightful day. And I would like to thank all the council members for your engagement and input on all of our conversation. And with that, Rick, I will ask you to close out the open session.
Great. And David, I'll thank you for the coordinating work that you've done. So with that, we will officially close out the open session of today's council meeting.